Hey, hi, hello. You know what? It's another really nice day today, and whoa. It shall not be filled with any negativity. It shall only be a positive day where good things happen. And we're gonna look at what the fuck is playing right now. Oh my god, I can't even. An explanation as to what? What are you talking about? I need my glasses. Oh, that. Yeah. Ignore that. Ignore all the tabs and whatever. What is this? It's a .gov website. .gov of another country. Well, that makes sense that it wouldn't load, right? Okay, well, Facebook, right? I got a little bit of food right here. It's the kind of food that you can just eat one-handed, very much so, you know, sandwich sort of thing, right? So, well, there's nothing much to this now, is there? Ekaterina Huglug. Okay. What is this? BenQ. Something BenQ. Where's with the BenQ? Okay. Oh, I'm digging this. Sausage. Not the biggest fan of sausage, but whatever. I mean it literally, okay? What is this? Yeah. Wow, you know? I can't read any of this. Okay. Wow. Okay. Yeah. And yeah. Wow. That's really fascinating. Yeah. I'm feeling quite fascinated right now with all of this. Yeah. Wow. Well, okay. So let's try to decipher the meaning here, right? So, well, it's like do's and don'ts, I guess, or something, maybe? This looks kind of like a projector, doesn't it? Yeah. Okay. I don't know why I was thinking like a... I don't know why... Well, I just go to printer right away, but... Oh, it's like a projector, right? It's gotta be, or something like that, yeah. I mean, digital micromirror device, yeah. Oh, I guess we could look this up and see for sure what this is, right? Like, this is just a manual in another language, I imagine they have. What? Yeah. That's not what I copied at all. That is not what I copied at all. Ooh. 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 Tube Screamer, huh? Overdrive Pro. Okay. MX808ST. Yeah, it's like that is just quite simply not what I copied, you know? Yeah, it's a projector, yeah. I mean, what else could it be? Looks like a projector. Is a projector. Yeah. Okay. So, you know, something with the manual or whatever. Read the manual. Okay, I guess. Plug it in. It has the sleepies. It's a sleepy projector. Something pertaining to, you know, like, say how it has, like, you know, sleep, shutdown, restart kind of thing, right? Like, you know, what's that? I don't know. But yeah, let's zoom in, right? You can go away, you know, but not entirely sure how we make you do that. It doesn't matter. Okay. So, reading the manual, I guess, and something relating to it, the device going to sleep, uh, getting flashed, um, like don't stand in front of it and stare into it or whatever as you're turning it on or something like that. Or just in general. Yeah. Mm, that's a really anime face right there. Yeah. And then, thumbs up gives you the go ahead to something. And then, that's even more anime than before. So, something pertaining to the light. Um, hmm. Hmm. Best guess, huh? Best guess. Hmm. Well, it could be. Yeah, no, I have no idea. I'm not the most familiar with projectors. I don't. What do you do with projectors, right? They project things onto place screens and whatever. Yeah. That's great. That's really, really great. So yeah. And then stop that. Stop that. Like, it's like anime slapstick humor or whatever. It's like, you know, this and then, well, that's just what you do next. You know? Good smack in the face or whatever. Or maybe it's to assert dominance. Maybe it's some kind of, yeah, thing. Okay. Why is your face just shifts around as needed, huh? As needed. It's almost like it's on a separate layer entirely, right? No, that couldn't be the case, right? Oh yeah, and then like it's it being your mouth, but you have your own mouth. It's so weird. It's so weird. And what's over, it's just that is so misaligned, right? It's just cutting right through your face, which wouldn't be that bad of a thing, I guess. Wouldn't be the worst thing in the world if we cut through the projector's face. The projector should not have a face, so. I mean, that's not how you're supposed to unplug anything. You're supposed to have your fingers closer to it, sure, but like, I mean, you're yanking the cord like that. Like, that's not good practice, and it's like, well, whatever. I suppose you could interpret this as, well, see, but it's going the other direction. So, I mean, it's like about dropping it, but then this implies like that you're going to spill this on it, but clearly it's not going to be spilled on this at all. They're going in separate directions. They're both going to fall off the table or whatever it is, but what's with the, if it's not going to, it's not going this, it's going the other direction, So what's with that? And then, along with the books, yeah. And again with the anime stuff, and I mean, sure, maybe this too, but to a lesser extent than this, and like being covered in, well, yes, just covered in books and like the lines coming off of it, the squigglies, and oh my. And this doesn't help at all either. Yeah, it's a lot better this way, I think. Oh, God. Well, I mean, yeah, this is obviously saying not to do this. Don't jab it with a screwdriver. Okay. Gee, I didn't know that. I didn't know I shouldn't jab it with a screwdriver. Anyways, this is like, you know, it'll get hot if it's covered with stuff, is what it is, yeah. Can't have good ventilation if it's covered with just whatever stuff. But yeah, for some reason, like, you don't really have that, like, back to back. You have it, like, here and here, and it's of the same thing, and it's like, it's all over the place, right? All over the place. I don't know, for like, I mean, its face disappears, which kind of implies that it's like for storage sake, you know, like where to put it, where to hang it up, and just kind of leave it be for a while. But then again, it's like, yeah, I don't know, because because its face is gone. I guess that's exactly what it is, right? That's what it implies. But same thing here. Well, it's just, its face is there, it's just not visible, you know. But anyways. Why would you put it next to it? Why would you do this? This should not have to be... Why would you do this? I don't know how I feel about the liquid droplets falling onto it either. Just that in the background. And what is going on in all these? Like, just, is it something with these language people? What are you guys doing? Why do you do this? What is this? You know, why? Upside down, or is it downside up? And that, there is no real, other than the, well, I don't want to say lizard or gecko or whatever it is. It doesn't really look like, its head is not like lizard shaped. I don't know. There isn't really a whole lot to go off of here in terms of, like, here, like, oh, there's a sink in the background overflowing with water and whatever. Which you have to, like, well, unless it's like clogged or whatever, you have to purposefully do that, you know? Which is like, yeah, just, yeah. And even, right, it has a little hole on the side all the time, like, you know, so you have to plug that up too, right? It's like, yeah, I mean, if the sink is overflowing, it's because you want it to, right? It's purposeful. You know, there's some deliberateness behind it, right? In a house with, what is this, like mountains and clouds, or with the line, with the, it's overheating, right? What the hell? ... I guess. Yeah, they're a little bit diamond shaped though, but it's not, it's off diamond, you know, like there's not, like, I mean, yeah, it's just the equivalent of whatever, but it's all of that, huh? And then it's just, here's what comes in the box. Oh, lovely. How is it that you make this weird too? Like, I don't know. Like, oh, I put my thumb on it and I pull it off, and it's to clearly display it or whatever, but like, I don't, well, you could just scroll down a little further and see it very clearly that it's a projector. Okay, makes sense. I just don't want to scroll ahead needlessly, you know? Wow, I mean, we're at the very beginning, huh? Wow. Yeah. I mean, you can just really about anything, huh? So, yeah, good ports. It has, it has VGA and everything like that, everything, everything a growing boy needs or whatever. Lock symbol. It looks like two misshapen wrenches. Everything looks misshapen. Yeah. Okay. What is that, like a wishbone sort of thing? Or lambda? Oh, actually, yeah. Well, no, I don't know. It's just you guys' weird symbols, I guess. Okay, well, alright. Kensington, HDMI dongle. Yeah. Okay. So, well, there you go. That's, that's really great. Yeah. And, yeah. Yeah. Okay. Yeah. We're looking for like, what transcends language, right? What goes past barriers, past beyond language barriers or whatever, right? Well, illustrations, of course, to some degree. I mean, a little for the most part, right? Aspect. I love aspect. I mean, yeah. So remote for the yeah. Well, of course, yeah. And what are we doing here? What's with the that is, wow. Yeah, I mean, okay. Huh. Okay. Sure. Yeah. You can display it this way, and you can also display it this way. Well, that's good to know. I wouldn't have known otherwise that I could do it both going to the right and going to the left. Maybe I would have been under the misconception, indefinitely, forever and ever, that, you can only do it one way, right? Well, well, yeah. Yeah, people all that all the time, whatever, on the ceiling. It just looks so stupid. It looks so stupid. And what are you just standing there for, you fucking alien? Oh my god. And and it can go both ways there too, and why are these like the same size, but then this one is bigger and this one is smaller? Why is this and why is that? And oh, and this is on the wall, and this is on the wall, and then this is from the ceiling, and this is from the floor. Yeah, I guess. I mean, you'd have to have something down here supporting it and whatever. Okay, well, anyways, whatever. Yep. Angular stuff. Yeah. I don't imagine this is numbers, though, or whatever, all of this. So, I mean, yeah. Okay. Well, this is the screen itself, right? That's what this is, and this is very 4:3 aspect ratio-ish, and then this is very 16:9, and okay, sure, why not? And there's the underside of it, yeah. I mean, at first glance, it looks like those are the feetsies. They're not. What are the knobs for? Who knows? Yeah, who knows? Something. They're for something. Yeah. Tighten and loosen and yeah. Well, that's what that's for. Focus. Focus, yeah. And then this is... Yeah. Based upon its orientation to the screen that you're displaying it on, right? If it's going upwards, going downwards, and you compensate for that. And if it's straight head on, you don't need to worry about all that. Yeah. Yeah. Of course you need that, yeah. Composite, this is not... There's composite. Yeah. You remember composite? The shitty speakers and... Oh, yeah. And your VGA and your DVI and your... God, there is so much going on here, So much going on here. Wow. Roku type shit and wow, wow, and God, what a mess. What a mess. What? Sure has a lot of ports. Doesn't have that many ports. We are just... Okay, well, and then it's plugging it into a separate thing, too, and that's why there's so much going on, right? And, yeah, it's plugging it into a bunch of things, and that's why there's so much going on, I guess. Yeah. Cable has two ends, right? Like, let's follow the pathway, right? It's going to a separate device thing, that you could utilize in theory or whatever, and this splits off into 3 from the VGA, and that's definitely not what it looks like, but for simplicity's sake, like a trident, yeah. And then this is a 2-way trident only, and there's another one there, and yeah. Wow, wow. That's really cool. And then audio weird stuff, and I don't know. Okay. Well, and then we have this. I like that. I really like that. Okay. That's a really short cable. For the sake of... Yes. Of course. Fit it all nicely into the frame and whatever. Yeah. I mean, there are the cables that are this short that do exist. I wonder what this is denoting. Because, it just makes me, think of, a little, ribbon thing coming off of it with, a little text on it or whatever. Okay, language. Yeah. English is right there, but, we're clearly... we're going off of the Japanese logic. Yeah, there is some of that sneaks in, right? Clearly in this image as well. Language, confirm, exit. I've heard of these words before. Yeah. Out of range, Yeah. And then we have this fucking mess. So, I don't know if this is just... Yeah, menu, back menu, yeah. Brilliant color. I'm very familiar with brilliant color. Yeah. Kensington. And then there's this. Oh yeah, of course, a little passcode or whatever, and there's settings, it's pretty clear, pretty obvious. Warning. What could the warning be about? Who knows? Yeah, more of that stuff. Set a passcode. Yeah, source. Switch your inputs. Why is there 2 separate ones? Because it's for 2 separate... Oh, well, 2 different kinds of devices. Right. They love to stick a bunch into one because they're too lazy to have separate ones or whatever that is. Whatever. Or too lazy or, to ship it with their products, right? Like, this has to match what's in the products, and in the products it has, You know, it has it all together, like ha 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 ha. You know, that's what happens with I don't know, one size fits all, but on a massive scale, you know, or whatever, or larger mass production. Yeah, that button. I like it. It looks stupid, and I like it. Yeah. Okay. Now we got some more of all this. Oh, thank you. Well, it looks a little bit off. I'll say that. That is, this especially, it's purple, you know. It all looks a little bit off, but yeah. You know. RGB CMY. Yeah. And there you have it too, but in the wrong order. MCBGRY. And then there it is. And well, why are we what? Anyways. Button stuff, something with the arrow pointing to, and then it'll look a little different on the two different devices, you see, because in this one it'll be just a button, and on this one it'll have little black bars on the side there. Yeah, you know, and it'll be surrounded by black here, you know, and the arrow is, the button aspect ratio is different, you know, and yeah. What? And why is it again? You know, we already saw all of this, and you know, this is really something. You know, this is really, really something. It's such a stupid sound. This, I don't know. You know what we had before of don't jab it with the screwdriver. Use it to turn the screws instead for its intended purpose. Yeah. What are we doing here, huh? What are we doing here? Well, taking off the top and how to unplug it and take it out. Yeah. That's yep, the little thing on the side there. Yep. Okay. And is this a display bit itself, huh? Or it's got a little handle, you know, a little, yeah. Pull it up and out, and then for sticking it back in, they really do hold your hand, huh? With these instructions, yeah. Well, it's better than, I guess, the alternative, maybe? I don't know. Cool. Yeah. And then, you know, well, you gotta screw it back, don't you? Don't you? Right? Yeah. Sure. Wow. This is a whole lot of something. This is a whole lot of nothing. This is a whole lot of, you know? Yeah. Wow. Okay. HDMI, HDCP, and 480i, 480p, 576i, 576p, 725 or 720 50p, 720 60p, 1080 50i, 1080 60i, 1080 24p. Ooh, cinematic, right? 1080 25p, 1080 30p, 1080 50p, 1080 60p. Anyways, I is interlaced and P is progressive, and you want to save 15% or more or something. You want to save 15% or more with progressive. Yeah. Stick them together. Why not? You know, they're lonely apart or something. 480i, 480p, 576i, 576p, 720 50p, 720. Wait, we're just repeating the same thing. Oh, this sucks. What is the difference here? MHL and then gibberish. More gibberish than just here where it's just gibberish and then with a dot. That is a proper dot, right? That's not a weird misshapen diamond dot. That's a proper dot right there is what that is. Component. Yes, I love me some component. It's the same thing. Okay, NTSC. Wow, I mean, it only goes up to 1080 60. Wow. Like all of that only for it to... Wow. Anyways, NTSC, PAL, SACAM, PAL M, PAL N, PAL 60, NTSC 4.43. Well, if this is NTSC 4.43, well, first of all, why is there no space here or a dash or anything like that? It's just shove it all together. Wow. Anyways, why is this NTSC 4.43? And then, you know, what is this one? This has got to be a different number, right? It's got to be. It's got to have its own number, right? Like, it can't just, you know, but they, you know, it's a given. Like, they don't tell you that. Like, that's just, maybe that complicates things, right? This is the 4.43 one, and this is the one without the 4.43. But it's got to be its own thing, too. What the fuck is the 4.43 even referring to? Sounds like an aspect ratio kind of thing to me, but probably not. And then that's it. That's it for 30 fucking minutes, huh? Yeah. Oh my god, it's my kindergarten teacher's name. Anyways, what a coincidence. A globular complex formation by MDA1 and the other 5 members. Non-disclosure agreement. Coincidence, I guess. And the other 5 members of the MCM protein family in fission yeast. Now, this is actually something pretty interesting, isn't it? Okay. Abstract, Department of Biophysics, Faculty of Science, Kyoto University, Anatomy, School of Medicine. We'll be here forever. Oh, you people, huh? You probably know your stuff, huh? Abstract, background in the fission yeast. Schizo. What? We're off to a great start. Schizosaccharomyces pombe, MDA1, MDA4, MIS5, and CDC21 proteins belong to the MCM mini chromosome maintenance protein family, which is thought to have 6 members. That's a lot of members. Each MCM member is required for the early stages of DNA replication and has a well-conserved central 200 amino acid domain containing putative? Okay. Putative ATP binding motif. I know what ATP is. Oh my god, I know what that is. I know what a few of these things are, okay. However, the precise molecular functions of MCM proteins are not yet clear. Results, we investigated the physical interaction of MDA1 protein with the other fission yeast MCM proteins using specific antibodies. Immunoprecipitation of... I hate this, like, just... Why can't there just be awkward, weird spaces in between? Just have it be like that. Immunoprecipitate. And you gotta follow it along to the next line. Oh, God. Of MDA1 protein leads to the co-precipitation. It's just so it looks neat on the sides or whatever. I mean, you could just, like, center it, you know, center it. Instead of it being from this side forward, it could be centered, and that would take care of your problem too, you know, or something. Like, I don't know. It's not gonna be neat, but, like, it's better than this shit. Way better than that. All the other members of the fission yeast MCM protein family, leads to co-precipitation of Immunoprecipitation of the protein leads to co-precipitation of all the purify the MCM protein complex by a combination of column chromatography. Okay, the neculative. What the fuck are you getting neculative from? Neculative. Is that a thing? Neculative. Ooh. Well, yes, neculative. Speculative. There is no recognized English word. I fucking hate you, AI. You should die. Kill yourself. AI. Fucking. Anyways. Yeah. Well, I mean, I already knew that. It's just neculative. Yeah. It's speculative that, you know what? It gets things wrong sometimes, so maybe it does exist, maybe, well, it has yet to exist, it exists now because I say it exists. Neculative is now a word, okay, because I just used it, I just came up with it, I just used it. It's probably you can come up with before, something like that, just not a word, not used. I say it is a word. I do declare that neculative is now a word. Wow, it's 6 p.m., huh? The purified fraction contained nearly equal quantities of the 6 MCM protein. Electron microscope observation. Oh shit, I'm skipping ahead. The native molecular weight of the MCM complex was estimated by gel filtration to the 560 kDa. Okay, the purified fraction contained nearly equal quantities of the 6 MCM proteins. Electron microscope observation showed that the MCM complex has a globular shape with a central cavity. Conclusions: we have developed a procedure to purify fission yeast MCM proteins in a native heterooligomeric complex form for the first time, which opens an avenue to further biochemical analysis. Okay. Introduction: DNA replication is a fundamental activity of biological reproduction. Oh, really? DNA replication is initiated by a recognition of replication origins by origins, a transacting positive factors that open the DNA duplex, providing entry sites for the factors that catalyze a further unwinding of the duplex in DNA synthesis. In E. coli, DNA A protein recognizes ORI-C and melts the duplex. Melts the duplex? Ooh, cool sounding. Bramhill and Kornberg, 1988. In eukaryotes, the mechanism for initiation and replication is less well understood than in prokaryotes or in bacteriophage and eukaryotic viruses. However, yeast genetics has become a powerful system for the identification of genes and their products, which control the initiation of replication in eukaryotes. Why do I understand like nearly all of that? Many chromosome maintenance genes have been identified as those genes which are essential for the initiation of DNA replication and constitute a gene family that can be classified to 6 members, MCM2 through to MCM7. The budding yeast, Saccharomyces cerevisiae, the MCM2, MCM3, and MCM5 genes have been identified as mutations which affect the stability of many chromosomes carrying an ARS and a centromere. MCM5 is identical to CDC 46, whatever, and heat-sensitive CDC 461 mutant cells cannot initiate DNA replication under the restrictive temperature. In MCM21 and MCM31 mutant cells, the frequency of initiation at chromosomal origins has been shown to be reduced, which leads to a delay in the S phase. Some of the MCM members show a strong genetic interaction with each other. For example, the heat-sensitive CDC 46.5 mutation suppresses a cold-sensitive mutation in the CDC 54 MCM4 gene, and the double mutant of CDC 46 and CDC 47 MCM7 is lethal. Hennessy et al. 1991. Dalton and Whitbread. Whitbread. It's like Blackbeard or something. Backbeard, right? If you got a hairy back or something. That too. Yeah. Exactly. Yeah. Anyways, okay, makes sense to me. Makes sense to you, makes sense to me. In the fission yeast, Schizosaccharomyces pombe, 4 members of MCM gene family have so far been identified as conditional lethal CDC mutants, and part of the MCM3 gene has been cloned by polymerase chain reaction PCR. CDC 20. You know, you guys with your wording, you make these things sound way cooler than they actually are. I mean, it's kind of boring shit in reality, but you know, have so far been identified. But your wording, you know, it's like, yeah, very science fiction-y wording, right? We're trying to make it seem so much more profound than it actually is. I understand that. So far been identified as conditional lethal CDC mutants, and part of the MCM3 gene has been cloned by polymerase chain reaction (PCR). The CDC21 plus gene is a fission yeast counterpart of budding yeast CDC54 MCM4, and the CDC21 mutant is defective in an early step of DNA replication that comes before the elongation stage. The cold-sensitive mutants of MDA1 plus and MDA4 plus genes corresponding to MCM2 and MCM5/CDC46 respectively. Now, when I see this and that and then plus, I mean, I'm thinking like, you know, it's positive, like atom, whatever, it's positively charged, you know, meaning it's missing at least one of its electrons that it, you know, for it to be whatever, because it's backwards, electrons are negatively charged, and that's, you know, kind of confusing, right? But and that's how it, you know, the flow of electrical current is yada yada yada. It's backwards. It's all backwards, yeah, because of that simple fact. Like, why the fuck do you have to, you know, why do you have to be negative, you know, and in your absence it becomes positive, you know? Just overly complicating, you know. And it's the same thing with with your goddamn brain. Like, pretty much everything, the right hemisphere controls the left side and the left controls the right side. Like, what the, and you were just, you know, flipping the image upside down so that you can then flip it right side up again with your eyeballs, and then all of that goes, you know, the occipital lobe shit is at the back of the brain. Like, we're going from the front all the way to the back, and we're feeding it through, and we're just switching it up. And why the fuck are we? That's just the way it is. Okay, I'm okay. So sure, supposedly, apparently, that's just like the optimal way of doing things. The needlessly complicated way of doing things, adding additional steps onto the thing. Apparently, that's just the optimal way of doing things. Okay, and that's just how it is. That's just how it goes. Okay, I mean, yeah, I mean, though that's not necessarily the case, because plenty of the time, that stupid fucking nerve thing, whatever, that wraps around your fucking aorta, it's like, you know, that's not optimal. That's vestigial structure type shit, right? That's whatever. Fuck you, okay? Fuck you. CDC21, where was I? I don't know. Elongation stage, right? The cold sensitive mutants, the genes corresponding to MCM2 and MCM5/CDC46 respectively, show an altered morphology of the nuclear. Golly, I am so stupid when I am tired, and I am always tired, and that is my problem. You know, I am so fucking stupid when I'm tired, and I am so not stupid when I'm not tired. And you know, it's just one of those, I mean, I don't even notice it, right? I mean, or you don't even notice it. Like, it's just, I don't know. I got good sleep, you know, I got good, well, you know, I slept for a few hours, woke up, slept for another few hours, but yeah, elongation stage. The cold sensitive mutants of MDA1 plus and MDA4 plus genes corresponding to MCM2 and MCM5 C46 respectively, yeah, show an altered morphology of the nuclear chromatin region and S phase delay under restrictive conditions. The CDC19 plus gene is identical to MDA1 plus. Forsberg and Nerd, yeah. Their studies or whatever is what this is referring to. The MIS5 plus gene, designated MCM6, has been identified as a mutation that shows a high frequency of mini chromosome loss. MCM genes are well conserved among eukaryotes, and interestingly, each organism has a set of 6 MCM family members. I mean, I guess it goes all the way along this, and then we go back up here, right? Yeah. Well conserved among eukaryotes, and interestingly, each organism has a set of 6 MCM family members, reviewed in Kiersey et al., 1996. Chung et al., 1996. The cell cycle-dependent association with chromatin appears to be a common feature of MCM proteins. In a budding yeast, MCM proteins are specifically present in the nucleus from late M phase until the beginning of S phase, and they are not detected in a post-replicative G2 nucleus. Hennessy et al. 1990. Fucking white bread. In higher eukaryotes, MCM proteins are detected constitutively in the nucleus, but their association with chromatin alters during the cell cycle. Todorov. Todorov is such a name, sounds like such a war criminal type name. The proteins are absent on metaphase chromosomes, but in the telophase they bind to the chromatin. The MCM proteins are displaced from chromatin during DNA replication, and interestingly, their dissociation from chromatin seems to correlate with their greater phosphorylation. Yeah. Light, right? This cell cycle dependent behavior of the MCM proteins is consistent with the putative. Well, okay, all right, putative, all right, not putative, but because it's on a separate line, you fuck, or whatever, fuck. You are a puta for putting the putative on two separate lines, okay? But the putative, okay, I'm going to say putative, fuck you, okay? Putative. It's putative, but putative. Replication and licensing factor originally proposed by Blow and Lasky. Replication licensing factor is a transacting positive factor required for the initiation of replication becomes inactive after replication is initiated, thereby restricting replication to only one round per single cell cycle. Indeed, it has recently been shown that the MCM3 homolog is a component of the replication licensing system in Xenopus. Immunodepletion of Xenopus egg extracts for MCM3 and inhibits the replication of Xenopus sperm chromatin and that of G2 HeLa nuclei. However, the HeLa nuclei of the G1 phase still replicate efficiently in MCM3 depleted extracts, probably because the MG1 nuclei have their MCM proteins associated with chromatin. Sure, MCM proteins share a well-conserved central domain of 200 amino acids, which contains the NTP binding consensus sequence. This central domain shows a weak similarity to the bacterial transcription factor NTRC. NTR. Oh no. Oh, NTR. Okay. C, right? The C. I don't know. Because of the R, the way it's coming out here. There's a little bit of additional space there. It's NTRC, right? It's not NTRC. It's NTRC. That's bad. That's really bad. Which catalyzes the formation of open complexes by RNA polymerase. This similarity has led to the suggestion that MCM proteins might be a DNA-stimulated ATPase that serve as a DNA duplex opener. However, the molecular function of MCM proteins has not so far been demonstrated. Using immunoprecipitation, several groups have recently shown that some of the MCM family members interact physically, although there might be some discrepancy between organisms. To understand the biochemical function of MCM proteins, we set out to purify MCM proteins from a fission yeast as an initial step. We have shown here that the NDA1 protein and MCM2 homolog co-purifies with 5 other members of the MCM protein family in a complex of apparent molecular weight of 560 kDa. Well, okay, K is kilo. What is DA? I mean, yeah, molecular weight. I mean, that's what it's referring to, but I haven't seen that before. Molecular weight is usually measured some other way, isn't it? I guess it is. Is it just this? I don't know. I swear I've seen molecular weight plenty before, and I've never seen no KDA. Maybe that's just me. I don't know, because maybe they just put the number but without the what it's referring to kind of thing, right? And for it to just be such a... That's also odd, too, for it to not be it's always a damn decimal and whatever. I'm probably just mixing things up. Examination of the MCM protein complex by electron microscopy is also presented. Results: immunoprecipitation of protein complex containing NDA1 and NDA4. Physical interaction of MCM proteins has been demonstrated by co-immunoprecipitation in several eukaryotes, including budding yeasts. In a fission yeast, we found that the interaction between NDA1 and NDA4 is sensitive to high salt concentrations. Data not shown. It is critical to choose particular types of salt solutions in which there are many different types of salts. Just as in there's several different types of all this shit, whatever. Anything that everyone refers to just as like one word, one name, and it's something chemical. There's multiple of them, and it's just there's the typically agreed upon one that we all see as that, and all the other ones we kind of just disregard. But like, you just fill in the blanks with some other thing, and it's the same format, same format. Therefore, there's different types of alcohols, right? There's a rubbing alcohol and there's methanol, and they're all toxic. But we found that the interaction between MDA sensitive to high salt concentrations. Data not shown. Okay. It is critical to choose particular types of salt solutions in which to observe the physical interaction between the MCM proteins. Yeah, and there's a whole thing there too, because so apparently potassium is kind of like an anti, regular sodium chloride kind of thing. So potassium chloride not only is a salt, but it also, like, if you have high sodium in your blood or whatever, it like counteracts that in a way. Apparently, like it just makes it so it gets more flushed out of your system quicker or whatever, something like that. I don't know, but potassium just steps in, and it's just like, so there's just stuff like that, and it's just whatever, I guess. Okay. As with everything, it's like, what is cheapest, right? What is cheaper? It used to be, oh, it wasn't so cheap for, whatever salt and spices and whatever, right? That's, now it's very, it's all dirt cheap, right? Aluminum used to be expensive. You remember when aluminum was expensive? More expensive than steel, I think it was, or it was very comparable, like just about the same at least. Steel is cheap too, actually. No, not steel. No, it was more expensive than steel, I think, right? Well, and then steel was really expensive too, right? No. Steel was... What? Steel pennies. Copper. I'm getting things mixed up. I don't know. MCM proteins, experimental... But aluminum was expensive at one point, right? Because of the particular something with its extraction, yeah, or whatever. Or its refinement, or... I think it's just its extraction, yeah. Something with its extraction, and then some nerd came along, and then whatever. Oh, now it's cheap as fuck. Okay. We'll just use it for everything. It's light, and it's this, and it's that, and blah, blah, blah. And there's so much all of it, just like, and then there'll be all these things of people touting all this and that about aluminum. Aircraft grade fucking, spacecraft type shit. Like, it's aluminum, okay? It is an aluminum alloy. It has got to be the most unremarkable thing ever. And we're saying aluminum like it's the next fucking thing. Like it's I don't know, just people, scientific marketing BS. I don't know, just aircraft grade aluminum. Blah, 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 blah. Do you even know what that means? I mean, yes, you know what that means, but it's like, it's this particular alloy. I mean, this is spacecraft grade right here. Okay. This alloy is a really nice alloy right here is what this is. This is a really nice alloy because it has aluminum in it. And that's strong, right? Aluminum is really, really strong, yeah? Anyways, but yeah, I mean, like, even steel, like, people always think this and that with steel, like, it's not that hard. Well, I mean, if it's just a little amount of anything, really, it's like, it's not, I don't know. You can break steel with your bare hands, Yeah, it's a little bit challenging or whatever, it's a little bit hurt your little fingies, but, it's not like, yeah. You don't have to be fucking, when it comes to like a chain with your bare hands, there's that fucking big muscular black wrestling dude, and they were supposed to cut the chain and they didn't, and the brr. So, just like, oh, I don't want to look weak in front of everyone kind of thing, so just, right? The chain is just a regular chain. It's no trickery here. It's not cut. It's supposed to be. It's supposed to be cut so that I can pull it apart like that, so that you're bending it instead of breaking it. Really, you're just... Well, yeah, I mean, you're breaking a little bit because it's like halfway through only, but, you know, you're more so just kind of like bending and twisting and contorting it, you know, versus like, yeah, I mean, whatever. But anyways, I don't know, semi-thicker chain, like, a little bit of reduction goes a long way, you know, of like, yeah, I don't know. You know, and the thing is, it's just, I don't know, just think horrific thoughts and you can do pretty much anything. Yada yada yada. Okay, Figure 1 shows the Kumasi. Let's not get into all that. Shows the Kumasi brilliant blue staining of SDS gels of amino precipitates with different antibodies. Protein A cephalose beads, which were loaded with anti-MDA1 antibodies, specifically precipitated several proteins that were not found in the precipitates from the protein acephalose beads loaded with normal rabbit serum. Xenopus sperm chromatin, normal rabbit serum, huh? Normal, you say. Normal. Okay, proteins corresponding to NDA1 and NDA4. Oh, we're continuing onto a separate page, but you're not without a proper break here. Well, yeah, because then it would be like halfway cut on this one. Yeah. NDA... Okay, so... several proteins not found in precipitates from the protein acephalose beads. Beads. Yeah, okay. NDA1 so yeah, proteins corresponding to NDA1 and NDA4, 118 kDa. NDA1 protein with a NDA4 NDA1 protein. What? I'm confused, because... something's up. Yeah. Okay. I don't know if... What happened? What happened here? Yeah, I don't know. Okay. Sure, yeah. What happened? I don't know. Something happened, I think, yeah. Okay, whatever. NDA1 Pro. We're just gonna... Okay. I got all of that. It's just how that connects to here is confusing me a bit. But I guess just... Because that doesn't make any sense. Right? That doesn't make any sense. NDA for NDA1 protein? Like, is it ND... Because those are separate things. Like, is it just that? Is it just like a little something missing or something in between there? I don't... Okay. Anyways, NDA1 protein with a hemagglutinin HA epitope and 6 histidine residues at its carboxy terminal end. NDA1 HA6 his KCO et al. 1986. Right, referring to some study or something like that. Something with some person. From the extracts of Ka25... I don't know. I could be wrong about a couple things like that, but like that, I don't know. I think it's just to be assumed like that's what it's referring to. It's referring to some study, some paper, some something, right? Okay. I don't care about any of all that. From the extracts of Ka25, we're not here for dick sucking of nerds, okay? The anti-HA beads. We're here for that. We're here for the cool stuff. Well, I mean, it's not really that cool. I don't know. It's okay, I guess. Extracts of Ka25, the anti-HA beads precipitated not only NDA1 protein, but also several other proteins, which included NDA4. Blah, blah, blah. Data from immunoblotting not shown, which were not detected when we used the extracts of 972 cells. Okay. Which did not express NDA1, HA6 lane 5. The protein specifically precipitated by anti-HA antibodies demonstrated similar electrophoretic mobilities in a SDS gel to those. I don't know what the hell a good bit of these things like stand for, but other than that, I mean, yeah, put it together pretty well, right? SDS gel to those found. Well, of course that sounds familiar. And it's 3 fucking letters like SDS. That sounds familiar. Okay. A lot of this sounds familiar for whatever fucking reason. I don't know. Maybe I'm just hallucinating, I don't know. SDS gel to those found in the precipitates by use of anti-NDA1 or anti-NDA4 beads. Compare lanes 2, 3, and 4. 5 protein bands were observed between NDA1 118 kDa and NDA4 80 kDa. In the precipitates with anti-HA beads, their intensities appeared to be nearly equal. Lane 4. And then there's over here NRS. I hate how this is displayed like this. Whatever. A. So I guess that's alpha or something. Alpha NDA1 or NDA4. I don't know. Yeah, HA. These both say HA. So that is what that is. Yeah. I mean, is it just 2 tests of the same thing? Well, no, because then it's also, right, then it's also showing NDA1, right, it's also showing that along here. IG, what is IG? IG, what is the IG? Okay. And the numbers are denoting... Yeah. 1, 2, 3, 4, 5. I mean, that's obviously just like, oh, well, there's a, yeah, Sure. Yeah, the lanes, yeah. Okay. Yeah, let's break this down. Let's break it down. Okay, compare lanes 2, 3, 4. Why do we hate lane 1 so much? Was there even any mention of lane 1? NRS. Why do you hate NRS so much? And what is Ka25 NDA1 HA6 is 972 WT? Golly. Yeah. That's what we're looking at. It's the purification of the MCM complex of S-POM-B. Yeah. 5 protein bands are observed, right, in the precipitates. Intensities appear to be nearly equal, right? Yeah. I just want to make sure I'm really getting this. Yeah, this is really important stuff. Yeah. Several proteins larger than NDA1 were also specifically observed in the anti-HA precipitates of the extracts Ka25. Compare lanes 4 and 5. Intensities, huh? Intensities, yeah. What is intensities referring to, even? well, I guess just how they react to light, right? Because you were mentioning that before. I mean, we're shining, right, electron microscope, whatever the hell, right? But is it just that? Or I might be missing something here. Yeah, maybe. I got most of it. Okay, several proteins larger than NDA1 were also specifically observed in the... Yeah. However, their association with NDA1 seemed to be rather weak, since they were not found in the purified fraction of the MCM proteins. See below. I will see below. Oh, that seems useful. Telling us what the fuck with figure 1, right? Immunoprecipitation of NDA1, NDA4, and their associated proteins. Well, I guess. Yeah. That is what's going on here. What about NRS? Why are we neglecting lane 1? This is so sad. Okay. I don't see a fucking mention of... Do we have NRS before? I can't even remember if we ever had a mention of NRS. What the fuck is NRS? What is NRS? We got Netarar, but what is NRS? I don't recall ever seeing NRS anywhere. Where is NRS? What is NRS? What the hell does it stand for? What does it mean? What does it mean? I mean, it'd be pretty easy to find, right? It's capital letters. You can't just chuck in, you know, and then we just don't even mention it. What is it? Like it's our control or something, I guess, or something like that. You could guess. Yeah, I mean, you could guess. I don't want to fucking be guessing. What is it? What is NRS? Wah. Ah, whatever. Fuck you too. Immunoprecipitation of NDA1, NDA4, and their associated proteins. Total extracts of Ka25, lanes 1 to 4, or 972 lane 5 cells were subjected to immunoprecipitation. Ka25 cells express the NDA1 protein. Right, and these are the Ka25s, apparently also including the... Yeah. How does that work? What was Ka25 again? I'd like to phone a friend. I don't have any of those. Okay, Ka25. Yeah. Well, I mean, yeah, down there, sure, but the thing with Ka25 is, yeah. The extracts of Ka25, like, just... Oh, hey, you know what Ka25 is? It's that thing right there. NDA1HA6 has... So Ka25 is just a simplified name for... But then NDA1 is also contained within that? You know? How does that work? And then NDA1 is also down here with this. It's like... That doesn't make any fucking sense at all. How can it be in 3 places at once? Like, just... You know? And this is the 972. WT is like by weight, but I thought you were... I mean, that's what I know it as, is WT is like by weight. But, you know, I mean, you make it sound like it's 900, right? It's 972 cells, and WT is, I don't know what the WT is referring to then. Something else other than that, right? It's not, I mean, why the fuck would we be, or why are we measuring the weight of them or something? Like, I mean, you can't even really do that all too much. Like, just, yeah, right, molecular weight, whatever, right, but, oh, that's what KDA is, yeah, okay. I mean, so yeah, well, I mean, we are doing that, it's just, but this is, But right, but that's not what this is, because, you know, and that's just the count. You know, that's just 972 cells. And but then WT, like, fucking, oh god. Oh god, oh god, oh god. Well, how can it be in 3 places at once? This is NDA1, this is NDA1, and this is NDA1. Ah. So it's like the NDA1 is present within things other than NDA1. And, well, I guess it's not over here, right? It's just... Or no, it is. Like, it's all along all of this, right? For each of these, right? This is where NDA1 is located. This is where NDA4 is. And then this is whatever IG means. Okay. Okay, I'm annoyed, but okay. Immunoprecipitation of NDA1, NDA4, and their associated proteins. Yep, that's what this is. Total extracts of Ka25 lanes 1 to 4 or 972 lane 5 cells. Yeah, this is okay, whatever. These are both HA, but this is HA within this, and then what even is HA again? Fucking god. I hate labels because they're so fucking, like, it's just letters and numbers, and like, what the fuck does that mean? You know, give it a proper name. Cephalose, I can remember fucking protein A cephalose. We just drop that immediately. Xenopus egg extracts, like, you know, but nah, fucking, I hate you, goddamn nerds. I hate you because you're so bad at everything you do. I swear to you're so bad with, like, naming things or whatever, okay? And stuff like that. All right? That's something you're really fucking bad at, okay? Yeah, I know, it's just like throwaway. They're like throwaway names, and like, I mean, we have to name a million, billion, trillion, fucking quadrillion things, so it's like, yeah, it makes sense, but it's like, come on, just like a second longer of just something, you know? Something, I don't know, something. Something other than fucking HA, NDA, fucking goddamn you. And all the stupid fucking present everywhere, stupid fucking acronyms that means nothing to me. Those are letters. I mean, you know, say at the top of the page, you know, what is that? What is that? Like, yes, MCM is fucking, yeah, I know what fucking, yeah, MCM is a fucking protein family. Yeah, oh, no shit, right? Fucking, oh god. But, you know, I don't know what the fuck HA is. I don't know what the fuck this and that is. How am I supposed to know unless I've read your other fucking papers, you know? I have no way of knowing. This isn't for me, like, this isn't, I don't know, it's on the internet, whatever, you know? You should be able to, you know, it's like a sequel or something, right? There are a million, fucking Kingdom Hearts timeline. Like, you should just be able to just jump right in and just pick it up right from here, you know? Like a legend, right? A guide, but yeah, like a legend, you know, like they have on fucking Globes or whatever. You know, just... It's sloppy is what it is, okay? It's lame as hell, and it's fucking, you know, fuck you, Yasuhisa Adachi, Jiro Ukura, Mitsuhiro Yanagita, fucking Japanese. I mean, that's why, you know, that's your problem. God damn. Fuck. Okay. Immunoprecipitation of NDA1, NDA4, and their associated proteins. Total extracts of Ka25, which is lanes 1 to 4, or lane 5, 972 cells were subjected to immunoprecipitation. Okay. Ka25 cells express the NDA1 protein that has an HA epitope. Yeah, okay, it's an epitope. What even is an epitope? What's an epitope? You know, I don't fucking know. You know, it makes sense that you would just know this shit beforehand or whatever. Explain to me, what is an epitope? What is that? You know, you mentioned it before somewhere in the sea of a million other words that are a little bit more interesting to read about than that, but, you know, epitope. Fucking epitope. E. coli. Fucking E. coli. God. It's easy to get E. coli and Ebola mixed up. I don't know. One is, yeah, you don't even, you know, Ebola is everywhere. No, E. coli is everywhere. Ebola is, forget it even exists kind of thing. Unless you, whatever, live in Africa back in like 2015 or something, right? But anyways, other than that, you know, forget it exists. It doesn't fucking matter, you know. It's like the Black Death nowadays. You know, oh, 10 people, whatever, with the Black Death every year. Oh, wowee, that's a lot. That sure is, you know, it's still around. Hundreds of years later after the plague, you know, it's still around and it's still bubonicking up. You know, like 10 people every year. Woo, watch out for the Black Death. You know, fleas on rats. Hysteria. Mass hysteria. Okay, cells are subjected to immunoprecipitation. Ka25 cells express the NDA1 protein that has an HA epitope. Okay. Sure. 6-histidine, which sounds like histamine. Histidine, I think you mentioned that before. Histidine, okay, sure, whatever. I don't know. I'm just thinking histamine. Okay, histamine tags. Tags, I mean, yeah, there's a whole thing, right? If we're fucking things with cells, I mean, yeah, they have all kinds of things like that we denote to, right? They're blah, blah, blah. You know, just like blood types, right? Whatever. They got all their fucking little things. They got all their little squiggly wigglies on the outsides or whatever. All their little things, you know, interact with each other and, you know, all kinds of gay sex and whatever. Okay. Well, anyways, whatever. 6 histidine tags at the carboxy terminus. Yep. Okay. Well, 972 wild type. So that's what it is. Did you say that before? I don't think you did. You know what I mean? Like, you just go on to do something before you even said what it is. Oh, now you decide to say what it is. What is WT? Oh, fucking, you know. Oh, wild type, huh? Oh, there you go. A rare instance of actually saying what the fuck it means. The stupid fucking letters. Oh, wow. Is that what it means? Wild type. Oh, wow. Wow. That would have been great to know before. Oh, fuck you. Extracts were loaded onto normal rabbit. Like, you should just have all this at the top of the fucking page. How fucking hard would that be? Like, god damn it. YMCA type shit somewhere here or something like that. Like, I don't know. I'm just whatever. This is why I don't read your stupid fucking papers, because it's stupid shit like this, like just needlessly all this garbage, okay? Extracts are loaded onto incompetence, okay, is what it is. Incompetence, all right. Extracts, I could do a much better job. Okay, extracts were loaded onto normal rabbit serum, NRS lane 1 or anti-NDA1 lane 2 or anti-NDA4 lane 3 or anti-HA lanes 4 and 5 antibody beads. Proteins bound to the beads were resolved on a 6 to 12% gradient gel and stained with Kumasi brilliant blue. Ah! Yeah, you do that with surgery stuff too, you know. Well, less so staining with a gel or, you know, blah, 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 but like, whatever. Oh, we're going to inject this into your bloodstream and it's going to, you know, like coagulate or whatever. It's going to concentrate in this particular organ or something. We're gonna see it, you know, we're gonna get to see the cancer all nice and good, and then we're gonna blast it away, and we're gonna take out a lot of the additional surrounding tissue just to be sure we really got it all, and that's gonna leave you a little bit scarred and deformed or something. Great. Really, really great stuff. Round of the beads were resolved in a 6 to 12. But anyways, yeah, I don't know. Just, you know, we can't do anything. We can't see it too good. I mean, well, you can see it too good. That's cool. That's cool that you can see it too good. Anyways, and then the way it interacts with things, and it's, you know, there's a particular thing, like it just binds to it nicely. It just sticks around. You know, it circulates all throughout your system, and you know, for most of it, it just kind of goes. What comes around goes around. But for some bits, for some parts of it, well, maybe it just sticks to it nice and good. You know, it hangs around there for a little while. That's cool, and you can just kind of. Visibility is nice and good with that. Oh, this thingy thing, this little dye that we inject in your fucking whatever. Oh, I mean, yeah, what amounts to that, but it's, I don't know, just chemical compound, whatever the hell. You know, it's harmless. It's harmless. It's harmless, they say. It's harmless. It's harmless. It's harmless. It costs $5,000 to inject you with this. Ah! Proteins bound to the beads were resolved, and probably more than that, were resolved in a 5 to 12% gradient gel and stained with Kumasi brilliant blue. Protein bands corresponding to MD, you know, always assume that it's more than that, right? Corresponding to NDA1, MCM2, SP, we're just throwing in a new thing there. It's to the power of SP. I hate you people. I hate you. I hate you. I hate you. Just throw new things in. Without anything of them, you know? And it's a big old gamble on if we're ever going to hear anything about what the fuck is SP? What is that? What is that? You can't just throw things in there. And I mean, yes, you can. That's exactly what you do. Fuck you. Okay, fuck you. Fuck you. NDA 1, MCM. Right, it's MCM 2. Okay. SP. What is SP? Like, it doesn't really matter. It's not the central focus, I guess, but it's like, I mean, you're referring to it here too and all that, and fucking white bread, Dalton and white bread. God damn it. Fuck you, white bread. NDA 4 indicated fat bands. That's literally what it says. Fat bands indicated as IG. Immunoglobulins. Oh, well, that makes sense. Sure. But then it's immunoglobulins are indicated as IG, not fat bands are indicated as IG. Right? Affinity, fur affinity. God damn it. Released from the affinity beads, the positions of the molecular weight markers are indicated on the sides as KDA. Okay, well, now you say what that is, right? I mean, yeah, I kind of semi-assumed, but, like, I don't want to fucking... Like, the KDA is very variable. Like, it is. That's just the case of the KDA is all over the place. Like, I didn't... Like, I mean, yeah, I could assume, like, that's what the word numbering all about, mostly, other than the 972 cells, but it's like... But at the same time, it's like, I mean, like, the KDA is just variable, you know? Its weight is variable. Yeah, weight changes all the time. Yeah, but, like... Well, if it's, like, its interaction with other things or something like that, like, we're adding weight onto... Like, we're adding 5 pounds onto the already present 55 or 15 and things like that, okay, I guess, but it's like... whatever. You know, and like I don't want to fucking make a mention of this and that of like what I think it might be, because it's like, well, I mean, that sounds stupid. If it isn't, then it sounds stupid, right? And well, of course I'm this way all the time. If it isn't that, then I sound stupid, you know. But hey, it looks, oh well, fucking just make a mention of, just make a fucking mention of it. Okay, well, I think that it might just be fucking the KDA, that might just be the fucking molecular weight, but it's like, what if it's not? What if it's not? So what if it's not? Anyways, okay, so that was that, and I guess it goes from here, but then, well, this is its own thing. This is a part of up here too, kind of, right? See below. I will see below, okay? Fuck you. 80 KDA were identified by Western blotting. Western, okay. Direction now. Now we're just throwing direction into the mix, like, or... Yeah, I mean, obviously, other things that Western could mean. It could be someone's name. It could obviously be a genre of fucking movies or whatever. But anyways, we're identified by Western blotting of a duplicate gel. Data not shown. Irrelevant, right? A very similar staining pattern was observed from precipitates with anti-NDA4 beads. Lane 3. Co-amino precipitation of NDA1 and NDA4 was unlikely to be due to the cross-reaction of the antibodies with MCM proteins, which share some structural similarity. The anti-NDA1 antibodies did not recognize NDA4 in the crude extracts, nor did the anti-NDA react with NDA1, although the anti-NDA4 antibodies do seem to cross-react with other MCM proteins. Wonderful. In this study, we use extracts prepared from the asynchronously growing strain Ka25, and that's what Ka25 is, right? It's just referring to the fucking strain. Underwhelming. I don't think you mentioned that before. Yeah, no, like it was just now that we're mentioning. Oh god. It's just the asynchronously growing strain. It's just the fucking strain. Like, yeah, I mean, that makes sense. It's just the fucking strain, but it's like, I don't know, I'm getting like hyped up for like, oh yeah, it's just, and it's referring to fucking, God, I hate this. Okay. See below, it expresses, in our study, which expresses, and then is that what this is? But it's like, but then this doesn't have a period here. Like, no, that would have to be this, but then like, which expresses, well, it's not continuing here. That doesn't make any sense. What does it go down here? What the It's like a manga with these people. Is it supposed to be read right to left? Because that's how they fucking do it, Japaneseians. God damn it. Like, God damn it. You see what I mean with this shit? Like, what the fuck? Which expresses, and then what, does it fucking go back up to the previous page or some shit? Like, what the fuck? It's expressed like it makes sense that it would be this, but this doesn't this hasn't ended. This is like continuing on to the next page, but that's stupid. It's all fucking stupid. Oh my god. It's all fucking stupid, okay? It's all fucking stupid. I mean, this is what you get when you get fucking 3 people on one paper, right? They don't even, you know, who fucking knows how whatever their English even is, like, God. God damn it. God. I mean, you guys even read your own fucking paper beforehand? You know what I mean? Like, you just write it and then just put it out. Do you even read your own paper before you publish it? You know? Oh, in this case, a document, of whatever, but like, I don't know. You know how fucking impossible it is to this and that? Like, okay, whatever. I don't know. Okay, see below. Let's see below about their weak association. They were not found in the purified fraction of the MCM proteins. Let's see below. Okay, sure, whatever. Who gives a shit? It's all out of order. Okay, well, I'm still getting it. It's just like not with your help anyway. NDA1 associates with all the other members of the MCM protein family. The results of immunoprecipitation have indicated that NDA1 forms a protein complex with both NDA4 and with some other proteins. Which ones? Which other proteins? Well, here we're mentioning some other things down here, aren't we? Well, that's cool. Okay, yeah. To examine whether other members of MCM protein family associated with NDA1, we collected antibodies against other members of MCM proteins in S. Pombe. Pombe? Wasn't that the thing with the eruption, the volcanic, whatever? Okay, anyways, nothing to do with that. We raised anti-Sera against the part of the fission yeast MCM3 protein encoded by the DNA fragment which had previously been cloned by PCR, hereafter designated MCM3SP. Okay, sure. We didn't have the hereafter designated as whatever for any of these things before, but okay. Or things like that. Like, it's just so inconsistent. I understand. It's just... Your paper is impossible to read, and I don't know. I'm just going to be a critic of it all. I'm going to critique your really, really bad capturing of your findings, your results, and your all this, okay? Because of how you're illustrating it is very improper. I don't know. It's not very good. It's not very good. You know? This is such fascinating stuff, and you know what? It's bogged down by the fact that you can't fucking speak of it all too good, huh? Can't speak too good. Okay, Coxon et al. 1992. In a fission yeast, the MCM7 CDC47 homolog had not been identified. We designated degenerate. Yeah, degenerate. Oh, I agree. Anyways, you got my attention. You know, you got my vote or whatever. We not designated. Anyways. We designated degenerate primers. We designed degenerate primers against the regions which were conserved between the CDC47 of S. cerevisiae. Whatever that, I mean, we know what that is. It's like along with S. Pombe, but like, okay, whatever. And the human MCM7, a whole bunch of this shit, whatever that's referring to, MCM7 in parentheses, P1.1 MCM3, well, MCM3 obviously, slash P85. What is P85 and P1.1? Who knows? Who cares? It doesn't matter. Okay, sure. Proteins like MCM7 and then in parentheses MCM3. I don't know exactly how this works, but okay. And then apparently this is like, it's both things. It's both MC. What is MCM7? Oh, well, you see, MCM7 is also CDC47, and it's also MCM3 in some particular way. Here's how it is, slash, and then in parentheses. Okay. Whatever you say, buddy. Fucking buddies, fucking Japanese people, perverts. God. Nagasaki and Hiroshima. Human protein C experimental is before your time, right? But maybe, maybe your grandparents or something, I don't know. you know, and then you wouldn't be picked for this, and it would be someone else, and they would be able to, you know, and then they would be able to write this shit better, you know. Yeah. Proteins, Dalton and white bread, 1995, of course. Yeah. S. Pombe genomic. I'm adding on syllables. I keep on doing this. Right. Whatever. Yeah. Dalton white bread. Okay. S. Pombe genomic DNA, 0.33 kb sequence. Okay. Kilobyte? No, it's not kilobyte. Well, what else would it be? Gee, I don't know. What are you referring to as KB? That's the only thing that comes to mind for me is kilobyte. 0.33 kilobyte sequence. Like, unless stated otherwise, what the fuck else am I supposed to know what it means? I don't know. Sequence was amplified by PCR. Well, you know, and that's the thing. PCR, at least it's, you know, PCR is better than NTR by a long shot. A fucking long shot, okay? Amplified by PCR, which, when translated into amino acids, show the highest homologies to the MCM7 proteins and other eukaryotes. Figure 2. No thank you. The sequence, where is figure 2 anyway? Like, this is figure 1. What is figure 2 on the next page? Figure 2 is on the next page. Oh my god, we're referring to some. Well, it's better to refer to something before it's the case than after it's the case, and then to do both is way worse, I will say. Oh god. Okay. Sequence was amplified by PCR, which translated into amino acids showed the highest homologies to the MCM7 proteins and other eukaryotes, which is down on the next page on figure 2. That's not on this page, that's on the next page. Okay. All right. The sequence was 78 to 85% identical to the particular domain of MCM7 proteins from humans. Arabidopsis and budding yeast, Springer et al. You know, I think it's so something humans. It's like you're referring to a different thing altogether. Well, it's stupid to say like, and then there's all that us and we and get away from me, okay? You're gonna science up my. Anyways. Arabidopsis and budding yeasts. The new sequence is also homologous with other members of the MCM family. However, its amino acid identity is reduced to 60%-ish. We produced rabid anti-sera against this peptide of the, and then it's past all of this. Fission yeast MCM7 homolog. You see how fucking, like, what the what is that? Fission yeast MCM homolog designated as MCM7 SP. Whatever. Did we get a thing with SP at any point in time? What the hell that's even referring to? I don't think so. Okay, it doesn't matter. Whatever. Designated as MCM7 SP, expressed in E. coli as a fusion protein with glutathione S transferase. The specificity of the antibodies were examined by immunoblotting of total cell extracts of S. Pombe Ka25, figure 3, lanes 1 to 6. Right. Not, it's on this thing, lanes 1 to 6. Okay. And then this is before that. You see where it was after before, now it's before. Like, you see what I mean? You know? Okay. Affinity purified anti-NDA1 and anti-CDC21 antibodies were recognized as single bands of 118 and 110 kDa, respectively. Line 6 and 4. Okay, let me. Okay. Immunoblotting of total cell extracts, which is just all of these. Okay, all of these. There's 12. Okay, well, it's all of these 6 down here. That's the specificity of the antibodies examined by immunoblotting of total cell extracts of S. Pombe Ka25. That's all of this, the crude extracts, right? Okay, got that. Affinity purified anti-NDA1 and anti-CDC21 antibodies were recognized as single bands of these weights, respectively, in lanes 6 and 4. Why are we saying 6 and 4 and not 4 and 6? Or respectively, right? Well, just refer to them the other way around. God damn it. Lanes 6 and 4. Ugh. As had been. Oh my God. Headache-inducing shit is what this is. As had been described previously, Ocasio et al. 1996, Majorano. Is that someone's name? Really? Majorano? And we still haven't read all this because of fucking... I mean, I was going on about the other things before. Majorano et al. 1996. Because in Ka25, the end... This is so messy. This is so sloppy. This is so everything. Because in Ka25, the NDA1 protein has an HA epitope and a 6-histidine tag at its carboxy terminus. The protein is slightly larger than the NDA1 in a parental wild-type strain. WT. Data not shown. Affinity purified anti-MCM7SP and anti-MCM3SP antibodies reacted with the 90 and 97 KDA proteins, respectively, although the antibodies showed minor... And then it continues down here. I fucking hate you. I really do. I really, really do. Okay, this is a bunch of... This isn't even... Is this a fucking image? This isn't even highlightable text. DNA stuff is what this is. Yeah, it's not gibberish. It's DNA stuff, but... the sequencer. What? Actually, no, what the fuck? There is just a bunch of other letters. What the fuck? HEDVKKALLVGGVD Oh, sorry. Let me start at the beginning. HEDV. Fuck you. Antibodies showed minor... Oh, sorry, it's way down here. We'll continue in a second, right? Golly. Okay. Figure 3. Yes, tell me all about this. What do you mean figure 3? What? I thought this was... What? Figure 2? Oh, is this... This is figure 2. Oh, my God. The plot thickens. Fuck. Okay, figure 2, comparison of the S-Pombe... Pombe, who gives a shit? MCM7 homolog with... I hate you guys and your labels. I hate it. Terms, words, things. Comparison of the S-Pombe homolog, and I just can't help but make fun of you for it. I don't know. Comparison of the S-Pombe homolog with MCM7 proteins and other organisms. The amino acid sequences of MCM7 from S-Pombe, S. cerevisiae, S. cerevi, fucking God. Human and Arabidopsis. Arabidopsis, not Arabidopsis. Hippity boppity boo, bippity boppity ba. Were aligned using the Clustal W program, Thompson et al. 1994. Asterisks indicate the positions where the sequences are identical. Of course. That's really great. Yeah. I can see that with my own 2 eyes. That's great. What are the dots? You say stuff about that. Okay, good. Very, very good. This is an improvement. I'm proud of you, son. You know? Dots indicate the positions where the amino acids of the 4 sequences are conservatively substituted. Yeah. Substituted. Conservatively substituted, right. Okay. Okay, yeah. And then figure 3 of this, association of NDA1 protein with all the other members of MCM proteins. In lanes 1 to 6, more mentions of lanes 1 to 6, but just, yeah, okay. The total extracts of Ka25 cells were separated on an SDS gel, as you mentioned before, and immunoblotted with antibodies against MCM proteins of fission yeast. The antibodies used for blotting are indicated at the top of the figure. Wonderful. We have whatever. I don't know. They all have an A. I'm alpha, I guess. NDA4, MCM7, MCM3, CDC21, MIS5, NDA1. And then it's the same thing over here, but just, IP with HA antibody instead of this is the crude extracts of it. It's the same thing for both, and it's in the same order and the same everything. It's just, yeah. Woo, it's, it's clearly, there's a little diagonal line, right? You can see a little pattern there pretty well, pretty easily, pretty good. Yeah. Okay. Total extracts of Ka25 cells were separated on SDS gel and immunoblotted with antibodies against MCM proteins of fission yeast. The antibodies used for blotting are indicated at the top of the figure. Yeah, right, I just read this. From left to right. Is it from left to right? Is that how we're reading, you Japanese fucks? You Jap fucks. All right, from left to right, anti-NDA4/MCM5SP, lane 1. Anti-MCM7, lane... Okay, we don't need to go through all this. Yeah, those are their names, and that's where they are. Antibodies, respectfully. Okay, now we're just going through the walking you through it, holding your hand type shit, which is completely unnecessary. Yeah, I see it right there. Okay, okay, what? All over the place. Lane 7 to 12, which is about their IP with HA antibody. In lane 7 to 12, NDA1 and MCM2 and its associated proteins were immunoprecipitated from the Ka25 extracts by anti-HA. Antibodies in the precipitates were subjugated to immunoblotting with the antibodies indicated at the top of the figure. Sure. Ka25 expresses an NDA1 protein as tagged with an HA epitope and 6-histidine residues. Residuals. Same thing, but anyways. Residues, yes, NDA1, HA6 his. Yeah, my favorite. The positions of the molecular weight markers are indicated at the sides as KDA. Yeah, sure thing. Okay. Got all that? Yes, I do indeed. Okay, so continuing on from here. Although the antibodies showed minor reaction. Okay, I guess it goes from here. All right. Left to right, it is. Apparently, supposedly. I guess with no deviation whatsoever from that. God. Showed minor reactions with several proteins. Lanes 2 and 3. Anti-MIS5 antibodies detected a 114 KDA protein, lane 5. Oh my god! And now it's on a separate fucking page. Why do you do this? Why do you do this to me? God damn it! Like, just, I don't know. Put a copy of the same thing on 2 pages or something if you have to. If you're still going to be referring to it. And then here's figure 4. Oh yeah, figure 4. This is figure 3. Okay. Okay. The antibodies showed minor reactions to several proteins in lanes 2 and 3. 2 and 3, right here. Okay. Sure. Anti-MIS5 antibodies detected of 114 KDA protein in lane 5, right here. Yeah. Affinity purified anti-NDA4 antibodies showed reactions with a major 80 KDA and a minor 100 KDA bands. Lane 1. We got a mention of lane 1 here, guys. We didn't really have that before, but now we got a mention of lane 1. Isn't that cool? Wow. The major 80 KDA band had been identified as NDA4 protein because the level of the 80 KDA protein increased in a strain transformed with a multi-copy plasmid. Multi-copy plasmid, huh? Okay, multi-copy plasmid. All right, I'm following you. All right. Okay, carrying NDA4 plus gene. Did you mention this before? Multi-copy plasmid? I don't know. You're throwing so many goddamn terms in there. It's like, whatever. I can remember multi-copy plasmid more than I can fucking whatever the fuck else, though, you know, so that, yeah, as I said before. Okay. Carrying NDA4 plus gene. Okisio, yeah. The minor band was probably a cross reaction with MCM3 protein, see below, and blah, blah, blah. Oh, see below, huh? And then where the fuck is this? Okay, this is continuing from the bottom of the page, huh? Yeah, really good stuff. Really good. Appeared to vary significantly, whatever. NDA1, HA6, HIS, and associated proteins were precipitated with anti-HA beads and subjected to immunoblotting with specific antibodies against the different MCM proteins. As shown in figure 3, all the MCM proteins were detected in the immunoprecipitate with anti-HA beads. We compared the Kumasi blue. Is that a brand? God, I'm surprised I haven't looked it up. Yeah, Kumasi. It's just, Okay, sure thing, AI. Thank you so much. I mean, or Wikipedia, either or. Yep, it is. It's a widely used anionic dye for staining proteins and polyacrylamide gels, and for protein quantification. Bradford essay. It operates by binding to proteins, forming a blue. Yeah, shut up. What the fuck? Oh god. Oh my god. Hey, squinty eyes over here. Okay. Nice fingerprint. Really nice fingerprint. Yeah. Looks just like the bottle stuff that you get with 3D printer. Yeah, the resin, the whatever. Yeah. Brilliant blue. Oh, that's really cool looking, actually. Yeah, that's really cool looking. I like that. I like this little line right here. Yeah. I mean, because all of it's pretty cool, but like, that's like especially cool how it refracts the light right there, along with the brilliant blueness. Wow, it really is brilliant blue, huh? Yeah, that's why we call it that. First used to visualize proteins in 1963. Hey, this is a really good dye and it's very visible. Oh, really? Man, I couldn't tell otherwise without you saying it for me, you know what I mean? Really? Gee. Well, anyways, no, I mean, there's something specific about it, right? There's absorbance, 595 nanometer, right? It's a little bit more than just that. It's just, here's it in solid form. Wow, that doesn't look very brilliant blue to me, you know? But then you stick it in some isopropanol solution, et cetera, some other clear thing that, whatever, works good for it, and wow, the brilliant blue. Oh my god, wow, so cool. Solubility in ethanol, slightly soluble. Really good stuff. Sodium salt. Yeah. What's with all the clipping, you know what I mean? It's like, and it looks like the fucking in The Incredibles with the goddamn, the black balls, and you got black balls all over you. You just, why didn't my 15% SDS page have any protein bands? I have quantified. What kind of a thumbnail is this? My god. This is what you get with nerds, right? Nicolay's. All right, sure. Let's see about Nicolay. Yeah, we're just in the middle. Yeah, sure. Why not? In the middle of it. Why not? Why the fuck not? All right, let's go on about. Yeah, let's see this and that. Okay, sure. Sounds good. All right, good enough.
Hello
right.
And welcome to Nicolay's genetics lessons. And today we have another case study.
Here, you want some subtitles or something? There you go.
Hello and welcome to Nicolay's genetics lessons. And today we have another case study, which I call "What's Wrong with My Gel Electrophoresis." So here's the topic of the day: why didn't my 15% SDS page have any protein bands?
I would like to know as well.
SDS page stands for the sodium dodecyl sulfate polyacrylamide gel electrophoresis.
Yes.
And here's a question. I have quantified my protein and peptides samples using BCA Pierce kit and applied 20 micrograms of the protein per well to run the electrophoresis. The molecular marker worked well, very well, but there were no bands from my samples. During the BCA quantification, I even had to dilute the samples because the stock quantification was too high, more than 2000 micrograms per milliliter, beyond the method sensitivity. Staining with bromophenol, blue, kumace, what could be happening with
it's brilliant blue. Get it right, damn it.
Gel. Any ideas? Do you think that silver staining could work better? Protein samples were diluted in water. I have found the answer in the question itself. Bromophenol blue and kumace is not the same stain. It is too...
Oh, well, there you go. I mean, right, it's easy to miss something like that, huh? Yeah. Well, I mean, because, yeah, I'm thinking, right, it's just another name for the same thing, but yeah, okay. There you go.
Bromophenol blue and kumace is not the same stain. It is two different stains which serves two completely different purposes.
There you go.
So this researcher mistakes
Easy to miss.
these two different stains, thinking that this is the same stain, just different names for the same stain, but this is not so. So hence there is no visible bands here. So what actually happened?
I mean, yeah, me
We see
too.
a nice
Upon just
bend
seeing
in
it, yeah, I'm fucking okay. We're just going along with it, right? We just kind of, right over it, just like that. Yeah. Oh, I mean, you kind of do just assume. Yeah. I mean, you must, right? It must be. It must be. I mean, but that's the whole thing with troubleshooting, huh? Not necessarily, doubt everything, right?
Ladder, because probably different dye was used here, probably the one that was in the kit, but we don't see any bands here because bromophenol blue does not interact with DNA or protein molecules which are being analyzed. It is simply aids
Useless.
in visualizing and tracking the process.
AIDS subtitles, please. No, don't capitalize it. He's not talking about that.
Of the electrophoresis run.
Although that is kind of, it's in the same thing, so it's like, whatever, right? It's in the same whole, I mean, we're looking at things of that nature, right? It's not completely 100% unrelated to all of this, right? But we're not talking about AIDS. Come on, subtitles. Come on.
So what for we are using bromophenol blue in gel electrophoresis? We added to the DNA or protein samples to provide visual tracking during the gel electrophoresis. It is blue color.
It is blue color.
And allows
so it is.
researchers to monitor the migration process of the sample through the gel. This helps to estimate the distance traveled by the sample and determines when to stop the gel electrophoresis itself.
Okay.
Bromophenol blue is also serves as a front migration marker, indicating the process of electrophoresis. It migrates ahead of the DNA or protein bands and can be used to estimate the distance migrated by the sample relative to the front. Bromophenol blue, being a smaller molecule, migrates faster than larger DNA or protein molecules.
Of course.
When the bromophenol blue
The same amount of energy.
has reached the desired position in a gel, it indicates that the gel electrophoresis run is complete and the gel can be stopped. Bromophenol blue also helps visualize the sample loading process. By adding it to sample, researchers can easily observe the sample entering the wells or loading slots of the gel. This ensures accurate and consistent sample loading. But again, because it doesn't interact with proteins and DNA,
it is useless.
We wouldn't be able to see any bands here. Of course,
what
if
a
we
waste.
use Kumasi Brilliant Blue or silver
the
staining,
good stuff.
we would be able to see those bands. Because we use
silver color or actual silver, because it really could go either way. I mean, with this kind of thing, it really, really, really could go either way. It doesn't matter, but just, I don't know.
Those stains for protein visualization. For example, Kumasi dye binds to proteins, resulting in a visible color change. After electrophoresis, the gel is typically stained with Kumasi dye to visualize.
Oh, look at that. It actually did it right with the dye this time.
Protein bands. So you still can use this gel to visualize those bands if you would use correct stain. So Kumasi dye interacts with various amino acids residues
you tried your best.
in the proteins, particularly basic amino acids such as lysine and arginine. We also can use Kumasi staining for protein quantification. The intensity of the stained protein bands can be compared to known standards or reference sample to estimate relative protein concentration. And of course, with Kumasi Brilliant Blue, we can stain bands and we can extract them from the gel for further analysis, such as mass spectrometry for protein identification or enzymatic digestion for peptide analysis. And my last note would be, I almost forgot to mention that bromophenol blue actually can be of the different colors as you see, and its color depends on the pH of the solution. So we also add bromophenol blue in our...
What doesn't depend on a lot of things, I guess. I don't know. What doesn't depend on the pH? Well, then it wouldn't be bromophenol blue anymore. No, it would still be bromophenol. Well, you'd have to add something in order to change the pH. Well, you have to or take something out or whatever, but add something in, and then it wouldn't just be bromophenol blue now, would it? It would be bromophenol blue and something else. You know what I mean? So it would be, it's affected by that thing, right? It's not just bromophenol blue anymore. It's not
buffer in order to control pH of the solution. So as you see, this color much about pH between 7 and 7.4, maybe somewhere around 7.3. And this is all for today. Subscribe and see you in the next video. Goodbye.
So I guess it's, it's blue over here at a more alkaline. So, okay. Yeah. Thank you so much. Why is sex a thing? Okay, please. Oh, no. Thank you so much. Wow. He's old. Anyways. Whom people perceive as more attractive 2 years. You know what? Why do we have whom people perceive as more attractive and then we have photos of children? Okay, what the fuck,
Hello and welcome to Nikolai's Genetics License, and thumbnail for this video is not a clickbait, but before I will move to us humans,
the thumbnail.
Let me...
What? What do you mean? Thumbnail clickbait? Oh, no. All right. Well, anyways. All right. Well, anyways, you were good for something at least, Nikolai. All right, you were good for something. You were good for, that at least, okay? All right. Neculative, right. Okay, so anyways, back to this. That's cool, I guess. Right? Yeah. Where were we? Who knows? Who cares? I don't know. Below in Nico CCO identified as protein because the level of the protein increased in a strain transformed the multi-copy plasma. Yeah. Well, I mean, I guess we were just at where we're highlighted, huh? Or something. Sure. NDA1, HA6, HIS, and associated proteins were precipitated with anti-HA beads and subjugated to immunoblotting with specific... No, yeah, I think we were down here or something, but yeah. It's shown in figure 3, back up there, with the 12, 6 and 6. All the MCM proteins were detected in the immunoprecipitate with anti-HA beads. We compare the Kumasi blue staining, figure 1, lane 4, and the immunoblots closely, figure 3, and data not shown. The 5 protein bands between NDA1, 118 kDa, and NDA4, 80 kDa in the anti-HA precipitates, figure 1, lane 4, were identified as MIS5, 114 kDa, CDC21, 110 kDa, unknown, 105 kDa, MCM3SP 100 kDa, and MCM7SP 90 kDa, respectively. See also figure 5, lane 8. I don't want to. The association of the unknown protein of 105 kDa with NDA1 might not be very strong because the recovery of the 105 kDa protein in the immunoprecipitates appeared to vary. And then we go back up here, very significantly, depending on the antibodies used. Compare figure 1, lanes 2, 3, and 4. Sure. The 105 kDa protein was lost in the purified MCM complex fraction. See below. And then I guess continuing on here. Purification of the MCM complex protein complex from fission yeast. To understand the molecular function of the MCM protein complex, we started by purifying the native MCM protein complex from a fission yeast. Throughout the purification, NDA1 an NDA4 protein. You know, I mentioned alcohol way before, and I mean, I'm obviously yeast is a part of that too, right? I don't know. It doesn't have anything to do with anything, but you know, I don't know. Okay. Throughout the purification, NDA1 and NDA4 proteins were followed by immunoblotting. The integrity of the MCM complex could also be verified from the co-fractionation of the NDA1 and NDA4 proteins. Total extracts were prepared from asynchronously growing cells by disruption with glass beads. The extracts were fractionated by centrifugation. 80% of the NDA1 and the NDA4 was soluble and was recovered in the supernatant. Okay. After centrifugation at 150,000 G? G as in G. You can do that? It goes up that high? That fast? This is the same thing as the other thing all over again with whatever the hell it was. Yeah, the fossils, the samples, like 150,000 G. What is that anyways in... Okay, so it's 9.81, is it? Right? On average. On average. On average. That's an average. Same thing with body temperature and whatever. And that's changed over like the past 100 years or so a good bit. Like it's you're cooling down and all that. Anyways, yeah, it's 9.81 meters. All right. I'm not... Yes, I could fucking calculate it myself. I could even do it in my head, but no. 9.81 meter. I don't fucking meters anything. Okay, and then 150,000, right? As simple as that, huh? Meters per second squared, like... Okay, now... Well, that's really nothing on the scale of anything. But what to do if you find a baby opossum? You know what? Sure thing. Let's see about this really quick. What to do if you find a baby opossum? Opossum mothers kind of suck. Yeah, there's a lot of animals that, you know, the whole abandonment thing. Human beings as a species are very needy, you know, very, very needy. And you can, you know, and you, whatever people, you know, you can try to act like you're not, but you are very needy. We are all very needy. We're a very needy species. Very, very needy and very useless in many, many ways that a lot of other species are not. Other species, another species, huh? Speaking of another species, what are you doing? Just like, I don't know, just in the kitchen. You're just sat up there on the fucking stovetop, just, you're mine now. Anyways, 3-month-old opossum who was separated from its mother is, you know, completely useless for like the first few years, just whatever. Who is separated from its mother is released into an outdoor enclosure for monitoring at the Cape Wildfare Center in Barnstable, Cape Cod, Massachusetts. Why humans don't have tails. Don't need them. Spring has finally... It's as simple as that. Just don't need them. For anything, really. Right? We're not blah, blah, blah in the trees. We don't need to be. Just don't need them. I guess. It's as simple as that. You know, it's usage wanes and whatever, just don't need it. Spring has finally arrived in the northern hemisphere, sweeping winter away with flowers, outdoor activities, and cute babies. Whatever. I don't think babies are all that cute. Typically, I think they're pretty fucking unsightly, unsavory, and I think they're... Unsavory is maybe not the word to use, but okay, like just... unsightly, but for things other than sight. All right, that's what I mean. While it's safe to assume that human younglings My hero. No. Okay. Anyways. We'll be outside in higher numbers. Of course, we're talking about baby animals. We are, huh? In the springtime, it's not unusual to find babies such as chicks and squirrels separated from their mothers. Yeah. And they're a good bit more so mostly fine and okay than Dora the Explorer sent me a, as she says, good article. You want to look at it? All right, maybe we'll look at it. Springtime, it's not unusual to find. Yeah, spring, anytime, whatever. I don't know, whatever. They don't really need their parents like you do, kind of thing. All too much. But the protocol for what to do about it, they're considerably less useless, all around. Yeah, I don't know. Anyways, but the protocol for what to do about it depends on the species and the female's motherly instinct. Whatever. In the case of opossum mothers, that instinct is comically low. If given the opportunity, squirrel and raccoon mothers will take their babies back, but these marsupial mothers are less forgiving. With possums, opossum, same thing. Shut up. We generally say that reuniting is not possible because mom generally has just kept going. It's trying to stick two positive magnets or two negative magnets together. Yeah, whatever. Just not interested. Bye. See you later. Stick them together and just ignore you. Yeah. Got other things to worry about. Sunny. Priya Patel, wildlife veterinarian at Massachusetts New England Wildlife Center, tells Popular Science. Ooh, it's popular. It's worth clarifying that while possums and opossums are sometimes used interchangeably, there are technically two different animals. Yeah, another way in which your naming scheme is dumb as shit, you fucking nerds. And here we're talking about Virginia opossums, Didelphis virginiana. Okay. Sure. Why not? North America's only native marsupial. Oh, yes. Virginia. Oh, yes. This one. Right. This is the only one that you'll ever see. Meh. Opossum babies weigh just 0.13 to 0.16 grams at birth. Wow, that's pretty small, huh? The newborns immediately move into the mother's pouch where they attach to her nipples and are quite literally stuck for 55 to 60 days, after which they reemerge out into the world. What? So they're birthed, and then they're just like, you know what? Never mind. They just umbilical cord themselves right back, huh? All right. Well, I mean, I imagine just like with their mouths, they just latch on to the titty and don't let go for 2 months. Okay. That's what I'm picturing it as. All right. An opossum is seen they have pouches, I guess. What the fuck is this? What's going on with... Anyways. I guess. Yeah, there's other things other than kangaroo. There's another thing too at some point, right? Yeah, they have pouches. Okay. All right. Okay. Sure they do. Fucking whatever you say, Bernstein Bear. I don't... Whatever. Whatever. Sure they do. Sure they do. In mom's defense, opossum babies don't have the clearest call for help. They make sneezing sounds. If she's nearby, she might pause to take them back, but Cash Patel, has only heard of one potential case of a mother looking for her lost babies. Well, I mean, yeah, you're not going to hear of too many cases when, it's like you're stalking them and your stalking of them disturbs them and you're blah, 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 blah. Unless they don't know of you, if they don't notice you or something, they don't notice you, senpai. As such, you should call a licensed wildlife rehabber. Yeah, we're going to say that about drug stuff too. We're going to call him a rehabber. Wildlife rehabber. You guys are wildlife, is what you are, you know. We'll call it wildlife rehabber. Wildlife hospital. Well, same thing, huh? If you come across one or more opossum babies on their own, in the meantime, don't try to feed them. Baby animals have a particular diet, and it could upset their gastrointestinal system. Ungrateful. Who cares? Ungrateful sons of bitches. Be glad you're getting anything at all. Could let you starve. Fucking God. Patel explains. Nah. No. Just leave them to die. Who gives a shit? You might not always be asked to bring the animal in for care. If the individual is around 10 inches long, not including the tail, it's usually grown enough to be independent. Uh-huh. Yeah, they're grown. You know, incest is silly with them, but when I do it, anyways. Silly and comedic, huh? Is what this is, yeah. Baby squirrels are here. Here's what to do if you find one. Have they ever not been here? Why some animals eat their babies. You can always have another. Conserve energy. Whatever. It's a simple calculus. This whatever is finite. You know. Yeah. I mean, yeah. Mm-hmm. They're cannibals. You know, yeah. They'll, yeah. Dogs and cats, too. Yeah. In general, cannibalism of offspring is super widespread, yeah? Yeah. Anyways. Jeju. Some fun things to do at Jeju. Right, okay, so if we're going off of 150,000 G, 1,471,500 meters per second squared. Now, meters, huh? I mean, that's still really nothing, of course, in the grand scheme of things or whatever. But, yeah, the allegations which he denies. No, I did not do that thing. Yes, you did. No, I swear I didn't. They thought this priest was poisoned, but I don't give a shit. Good. Poison all the priests. What was it? The truth was so much weirder than him being poisoned. And then it's like 15 plus pages of teenagers and younger kids are learning coded predator phrases like map online long before they're... I don't even want to fucking... No. I much prefer the strains of whatever. So... meter... Okay. Light speed. Okay. Let's say light speed. I mean, of course, it's nothing like... Well, I mean... no, it's nothing even. Lights... just... Whatever. Yes. Give me that. Okay. Yes, I know. It's... Yes. It's 300 million. So you're telling me about a 1% of the speed of light, you know? Is what that is. Yeah. Yeah, I mean, it seemed like it was something like that. That's in a centrifuge at about over 1% of the speed of light. Okay. If you say so, if that's... I mean, it's really small stuff, right? I guess. Just over 1% of the speed of light. Okay. Alright. Sure. Fucking... But yeah, no, you're probably just referring to RPM, aren't you? Yeah. Which is... But I mean, you say G. In what world does G mean RPM? G means G, you know? Okay, well, based upon this shit, it's like, I don't know. Yeah, it doesn't matter. It's just, like, I mean, it's kind of unbelievable for it to be over 1% of the speed of light in the fucking centrifuge. Like, I don't know. Yeah. Well, okay, I mean, regardless, I guess it probably still is that, huh? I mean, you can calculate that too based upon, you know, its diameter there, what the RPM would be regardless, but RPM and G is referring to two different things. I don't care enough. See, that's the thing. I don't care enough. Okay? I don't care enough. The solubility of NDA1 and NDA4 did not change, because it doesn't matter. I don't know. Did not change significantly when the extracts were, it's really fast. Regardless, it's really fast. When the extracts were prepared from CDC10 cells arrested, jealous. And this is G1 phase, or from cells arrested in the S phase by hydroxyurea. I know what hydroxy is too, but anyways. Data not shown. The MCM complex dissociates. Know all about that too, but it's not referring to that. In a high salt concentration, and we noted that NDA1 and NDA4 were eluted. Whatever that is. Eluates, in differing fractions during chromatography. Yeah. Gel filtration of knee column eluates. Figure 4, the molecular weight of the MCM complex is estimated to be 560 kDa by gel filtration. Total extracts were loaded onto an affinity column. At NDA1, HA6 his and its associated proteins were eluted by imidazole. The eluates, they were on page 5 of 13, you know. Were fractionated on a gel filtration column, Superdex 200. The fractions from the column were separated by SDS page. The upper panel is the silver staining of the gel. Okay. Positions are indicated at the top of the panel where the molecular weight markers are eluted in kDa. The lower panel is the immunoblotting of the duplicated gel with anti-NDA1 and anti-NDA4 antibodies right here. Okay. So top panel is massive, and then lower panel is this tiny little whatever. Sure. Antibodies. NDA1, MCM2SP in parentheses, and NDA4, MCM5SP in parentheses, were eluted as a 560 kDa protein complex. The positions of the molecular weight markers are indicated on the sides as kDa. This right here, yeah. This looks like a polygraph test or something, you know? The little spindly, whatever. Kind of does. Right? No, it doesn't. I don't know. I'm trying to make a connection where there is none. Okay, and then continuing on. Okay, we're going to start over here. The MCM complex dissociates in a high salt concentration, and we noted that NDA1 and NDA4 were eluted in differing fractions during chromatography. With either a mono-Q anion exchange column or with a hydroxylapatite column, data not shown. We therefore chose resins which did not require a high salt concentration for elution. The 150S extracts with the, whether it's 150,000 RPM or 150,000 G, centrifuge. Extracts were prepared from cells expressing NDA1 HA6 his and were loaded onto an Ni affinity column, NDA1. I'm getting tired. NDA1, HA6 his, and NDA4 were co-eluted in 200 lowercase m, capital M for some reason. Error, I guess. 200 millimeter imidazole. Does that make sense? I don't know. Maybe it's referring to something else, right? It's milli, and then the M is referring to something else other than whatever, I guess. What is the M referring to then? Yeah. Imidazole, data not shown. See, Okisio et al. 1986. Okay. A small aliquot of the eluates from the Ni affinity column were fractionated on a gel filtration column, Superdex 200, 1030, Pharmacia, to examine the native molecular weight of the MCM protein complex, figure 4. And this is figure 4. The NDA1 and NDA4 were co-eluted between the marker proteins thyroglobulin 669. I've heard all about that globulin shit because my mom's fucking working, whatever. Anyways, which also I guess was my whatever at point in time too with janitorial stuff. Anyways, 669 kDa and ferritin 440 kDa. The molecular weight and Stokes radius of the complex was estimated to be 560 kDa and 76A with, you know, the little weird thing on that, whatever the fuck that is, respectively. Siegel and Monty, 1966. Going way back to 1966. These values are in good, which I mean, yeah, the thing was in the 60s too with the Brilliant Blue, its first usage. Oh man, Brilliant Blue, huh? Brilliant Blue, you know, a Brilliant Blue. Same thing with, you know, in a fusion reactor and fucking whatever, and it's because of really cool stuff. Oh, it's on a technicality. It's exceeding the speed of light on a technicality because the water surrounding the reactor slows down the light enough. So on a technicality, the radiation coming off of it is exceeding it because it's slowing, etc., etc. It slows it down so then it exceeds it. Then you get that Brilliant Blue. Yeah. Yippee wahoo. These values are in good agreement with the estimates of the size of the MCM complex in other organisms. Chong et al. I don't know, it's really cool as fuck is what it is. But anyways, Chong et al. 1996. Su et al. 1996. We noted that several proteins showed exactly the same elution profile as NDA1 and NDA4. Yeah, good agreement. Estimates of the size. Okay. For presumably they were other members of the MCM, although the fractions containing NDA1 and NDA4 were fairly pure on a small analytical scale, figure 4. A large-scale preparation using a gel filtration column of A1.5 m, whatever m is, could be millimeters. I don't see what else it would be inherently, but yeah. It's obviously not meters. Well, then this is saying centimeters here, so fucking maybe it is just meter, 1.5 meter, whatever. Biorad, huh? Yeah. 2.5, 67 centimeter instead of Superdex 200 appeared to contain more contaminating proteins. Figure 5, lane 5, and you're referring down to figure 5 now, right? Good stuff. Figure 4. You want me to go back and forth between the two? Appear to contain more contaminating proteins. Figure 5, lane 5 is this one, LU on the gel filtration, the second one on the gel filtration. So what are we comparing? Contain more contaminating proteins instead of yeah, compared to on figure 4, is it? I guess, because you were just mentioning of that. I guess so. Don't know. Okay, for further purification, we performed immunoaffinity chromatography, whatever, utilizing the HA epitope tag at the carboxy terminal of NDA1 HA6 his. I performed that utilizing the epitope tag. Okay. His, and then it doesn't end. It doesn't have a period here. You fucking assholes. Oh my god. You know, just, yeah. You forgot to put your period or whatever. You forgot to end your sentence. Again, I guess it just continues here. The gel filtration fractions containing NDA1HA6 his were pooled and loaded onto a protein acephalose column, which had been loaded with anti-HA antibodies. The NDA1 and its associated proteins were eluted by peptide competition as has been described previously. Field et al. 1988, Zhuo et al. 1992. The presence of 500 mm sodium glutamate reduced the non-specific binding and also increased the elution efficiency significantly. Oh, that's good. Without breaking the interactions between MCM proteins. Yeah, that's very good. The final fraction contained nearly equivalent amounts of 6 MCM proteins of fission yeast in lane 8 here. LU, the third one on the immunoaffinity. 2 bands of 58 and 60 KDE were also found in the final fraction. The portion of the MCM proteins in the final fraction was estimated from gel scanning to be... my bad. Oh. No, this is the same thing. Okay. Problem solved, solve your problem. Where were we? We're on page 6 now? Yeah, I guess so. Estimated from gel scanning to be around 84%. That's a weird way of around with the two things. What's the difference between it being 1 and 2? I usually just see it with only the 1. Is there a difference there? Like around on average, is what it is. But like, is there a difference between it being 1 squiggly and 2 squigglies? It's called a tilde, but I don't know if it's called a tilde if you're doing it like that. But well, and we actually did have that before. We had that on the image, right? And that's what that was, I guess, or some. No, that was on the that was on the cable thing or whatever. That was on the cable thing. You know, I had like this kind of thing, or maybe it was just that, I don't know. I'm putting it together. How does it relate to that at the beginning? I don't know. Anyways, all the MCM members have a modified form of the ATP binding motif, Kunin 1993. However, the ATPase activity associated with our purified MCM fraction was very low under the conditions we used. 1 ATP molecule was hydrolyzed per MCM complex in 30 seconds. See experimental procedures for details. We did not detect any DNA helicase activity in the purified MCM fraction, even with substrates that have forked structures. Data not shown. See experimental procedures. Okay, wherever that is. Morphology of the MCM protein complex. We examined the morphology of the MCM protein complex by electron microscopy after rotary shadowing, and then it continues on to the next page. It just fucking over all of this. So yeah, I know. It's stupid. Yeah. Figure 5, purification of the MCM complex of fission yeast. The MCM complex was purified from the Ka25 expressing epitope tagged NDA1 HA6 his, mention of that again, through the combination of Ni-NTA affinity column lanes 1, 2, and 3. Gel filtration column A1.5M, biorad lanes 4 and 5. And anti-HA immunoaffinity column chromatography, lanes 6, 7, and 8. The SDS gel pattern stained with Kumasi blue is indicated. Where? Indicated where? Where is that indicated? Yeah. Where is it indicated? It is indicated. It is indicated, huh? Where is it indicated? Gel? No. What? Okay. Whatever. For each chromatography step, the loaded sample lanes 1, 4, and 6. The first of each of these. Yeah, the low. The flow through 2 and 7 here and here. We don't have a flow through on gel filtration for some reason, I guess. I don't know what the reason. For some reason. I don't know why anyways, for some reason we don't. And the eluates, lanes 3, 5, and 8, the last of each, were separated on an SDS gel. Okay. The final fraction, lane 8, the third of amino affinity. Final fraction contained nearly equal amounts of MCM members that are indicated at the side of the panel. Right there. Yeah. Okay. I should probably make my mouse cursor more visible or something, huh? Yeah, I mean, maybe. Where was that? Yeah. Let me, where was it? It's, well, I was something with that before, but how do you change the color again? What is, that's somewhere, yeah. Just, no, but there's a color thing. Where's the color thing? It's not that. Look, it's my fucking email. God. I don't care. Whatever. I don't care. I don't care. I don't care. You see how little I fucking care? I don't care. Yeah, this is lame as hell. Enable pointer shadow. Oh, that's what I need. I need a pointer shadow. See what it looks like. It doesn't really look any different to me, but okay, sure, whatever. Why not? Oh, well, yeah, okay. Windows, change mouse, cursor, color, change it to a brilliant blue, maybe purple. I have this and that before, I just, accessibility, sure AI, whatever you say, okay, I'll listen to you, you stupid fucking. So it is just here somewhere, huh? Accessibility, yeah. Mouse pointer and touch. There it is. Yeah, well, yeah, no, this is, style. The stupid trails. Yes, but this is not, oh. Never mind. Yeah, and then the, well, I don't know, just for visibility's sake, whatever, you can see it good, right? Blue, I don't know, make it purple, maybe, I don't know. But that's not as visible even, you know, so it's like, you know, why would you do that, right? Does that, what the hell is going on with that? It just looks weird in a tiny window is what it is. For visibility's sake, and also just because I like blue. I don't know. It's cool. Yeah. What is this? Yeah. No. It needs to be exactly, it needs to be 164 or whatever. Why won't you let me fucking change it? Why don't you let me? Why do you have this here if you don't let me change it? What the hell? This sucks. There is no 164, not 264. There. Whatever. Yeah, it's a little bit of a deeper. Does it need to be? No, it's not that it's I mean, if you really want visibility, right, but or yellow. I don't know. Yellow can be stupid, but I don't. No, I don't. I just want my 164. Okay, is what I want. All right, because 64, and I like 64. Okay. Golly, whatever. Fuck you too. Okay, whatever. Can you see it good? Yeah, maybe a little bit better because it's not white on white. Staring at a white screen all the live long day. Yippee yahoo. Turn down your brightness. I don't know. Purification. Nobody's gonna fucking see this shit anyways. But anyways, purification of the MCM complex of fission. Nobody gives a shit about all this. MCM complex was purified from the Ka25. I barely do even, you know. Yeah, where was I? Gel filtration. I was down here. Gel pattern stained with Kumasi blue is indicated for each chromatography step. The loaded sample, yeah, is down at the very bottom. The final fraction here contain nearly equal amounts. Position indicated at the side of the panel, right there. these 6 little markers right here, 3 and yeah, indicated at the side of the panel. Positions of the molecular weight markers are indicated at the side in KDA. Right there. Yeah. Okay. We examined the morphology of the MCM protein complex by electron microscopy after rotary shadowing. Figure 6. Oh no, this is figure 6. That's cool looking. I like this. I like that. Yeah. Okay, figure 6. Most of the structures had a round shape with a... No. Examine the morphology of the MCM protein complex by electron microscopy after rotary shadowing. What appears to... No. Oh my god. No. It just cuts off. Okay. Figure 6. Most of the structures had a round shape with a mean diameter of 27 nanometers. Nanometers. Figure 6, panel A. This one, the first image here. Note that this value is an overestimate because of the contribution of the platinum coating. The rest of the structures observed were smaller and heterogeneous in their sizes. A closer examination of the particles that had relatively fine details indicated that the particles often had slits or a pit and appeared to consist of several domains. Figure 6, panels B to F. Panels B and C show examples of the particles separated into 3 or 4 domains. Stereo photographs in panels D, E, and F show an example of a particle that has a cavity. D, E, and F. Yeah. Is this just the same thing twice for no real reason whatsoever? I guess. Guess so. No, there's tiny, tiny differences. Is it just 2 images of the same thing, I guess? Like we took these two electron microscope shots of it just for the sake of it. Yeah. Okay. Maybe so. Whatever. Has a cavity, yeah. Stereo photographs. Oh, right. Okay, panels B and C show examples of the particles separated into 3 or 4 domains. Right here. 3 and 4, I guess. Stereo photographs in panels D, E, and F show an example of a particle that has a cavity. D, E, and F. Cavity, I guess, in the middle or something. Yeah, something like that. Okay. The cavity appears to be open to one side of the globular structure and goes fairly deep into it. We occasionally observe structures which consisted of a globular head with a short skirt. Lovely. Panel G, this one. Globular head with a short skirt, huh? That is not how I would describe this at all, but okay. In this example, in stereo, a pit can be followed from the globular domain to the skirt. This image perhaps represents a structure tilted on the surface. You wait for me. Okay, figure 6, observation of the purified MCM complex after rotary shadowing by electron microscopy. I know. The complex has a globular shape, panel A. Okay. I guess, and seems to be separated into several domains, panels B and C. Panels D to G, from here all the way to here, well, these 4, are in stereo and show that you're just repeating yourself, are in stereo and show that the complex has a deep cavity at the center. Sure, I guess it does. I don't... Or is this the Is this the... Are these holes or something? Is it inverted? Are white holes in the otherwise, like... Because you say cavity. Cavity, I think holes. Is that what that is? Like... I don't know. It's just, why would you do that, you know, if that is the case or anything like that? deep cavity at the center. I mean, like, just this or whatever in the middle there. But, like, why? Panel G may represent an image of a slightly tilted complex. May represent slightly tilted, like on your behalf, or what? Slightly tilted. How did it become slightly tilted? Bar 200 nanometers for panel A or 50 nanometers for the other panels. Yeah, it's more zoomed in. Woohoo. Yippee. Yeah. Okay. Fission. Okay, yeah. Discussion. We have purified a protein complex containing NDA1, MCM2SP, and its associated proteins in the fission yeast S. Pombe. The purified complex contained nearly equivalent amounts of all 6 members of the MCM protein family. Figure 5, lane A. All the way back up here. Lane 8, I mean. Sorry. So the last one here. Yeah. I'm not sorry. Sorry for saying sorry. Physical interactions between MCM proteins have been reported in Xenopus, human, Drosophila, and budding yeast. A recent reports, plural. A recent report. You can't just say a and then fucking plural it up. Like, yes, you can. You just did. But Japs, dirty Japs, demonstrated the co-immunoprecipitation, again with the fucking, okay, off the line of 6 MCM proteins in Xenopus. Our results are consistent with the results from Xenopus and have clearly shown... What is Xenopus? What is Xenopus even? Just... and have clearly shown the physical association of NDA. I mean, from what we saw in the first fucking page at the bottom or whatever, it wasn't even the first page, was it? It was the second or something. Yeah. Xenopus egg extracts. Okay, what is Xenopus? Of all things Xenopus could be, it's frogs. They have no neck. Neckless frogs with claws, apparently. Yeah. 20 species are currently described within it. Okay. The African clawed frog is one of the most studied of all amphibians. It has no neck. Frogs are supposed to have at least somewhat of necks, right? Yeah. Well, sometimes not, huh? Well, I mean, they still have necks. It's just not clearly visible. Like, they can still move their fucking head around a little bit independently of their stupid fat-ass body, but whatever. In its lifetime, a female Xenopus can produce as many 30,000 eggs, depending on the particular species, and lay thousands of eggs each year. How many of those hatch and how many of those, you know, live all that long at all? You know, not many, huh? Can be bred and maintained easily in the laboratory. It can live for more than 30 years. Single one of these damn frogs for more than 30 years, huh? Tiny ass little fucking things. More than 30 years, yeah. That's usually not the case with small things, right? Small things living longer than other things, you know? 30 years. 30 years, yeah. Laboratory, whatever, it doesn't matter. 30 years is a long time for a tiny ass fucking frog. How about that, huh? The frog is genetically surprisingly similar to humans, which means that scientists can model human disease in this amphibian and replace the use of higher sentient species. It's a rat analog. Am I using that word right? I don't think so. Okay, yeah, it's some stupid fucking company. It's always some stupid fucking company. Analog. No, not analog. Analog adjective. That's as an adjective? Huh? comparable to another. I mean, yeah, sure, whatever. Okay. Although the eggs are initially opaque, unlike humans and other mammals, the xenopi, no, okay, apparently not. Offspring grow outside of the body. Yeah, frogs, tadpoles. This is not specific to Xenopus, whatever. Outside of the body, although the eggs are initially opaque, within a couple of days they become transparent, enabling researchers to see how the tadpole grows at all stages of development. Like this isn't the case with other frogs, too. Like it's specific to these, huh? Well, gee, I don't know. What the fuck do you know about frogs? Apparently nothing. I don't know. Okay. Bye. Back down to page 6 or whatever. Well, I was past all of this. Page 7 of 13. We're getting along there. We're over halfway. I'm tired. Okay. Fairly open to one side of the globular structure and goes fairly deep into. We occasionally observe structures which consisted of a globular head with a short skirt. Panel G. Purify MCM complex. Whatever. Oh my god. Where were we? We're past all this. We were on page 8 actually now. Fission yeast S. Pombe, the purified complex contained nearly equivalent amounts of all 6 members of the MCM protein family. Yeah, It was at some Xenopus. Okay, I guess it was here, right? This was the mention of Xenopus, which prompted. Okay. Physical figure 5, lane 8. Physical interactions between MCM proteins have been reported in Xenopus human, Drosophila, and let's see what this is. And budding yeast. Well, we know what budding yeast is, obviously. Fucking yeast, but okay. Drosophila. I don't want to just assume budding. Doesn't budding means reproducing? Yeah. Wow. Anyways, yeah. Okay. Fruit flies. All right. Stupid motherfuckers. Understood. Yeah. What is HG? Let's take a look at something else for a sec. Really quick. Is this going to load? Ministry of Communications. Digital Egypt. All-encompassing. Oh, I love that. All-encompassing. Yes. All-encompassing vision and plan laying the foundations for the transformation of Egypt into a digital society. Good luck with that. Decent Life Presidential Initiative. ICT. Wow, you guys. ICT higher education ministers discuss cooperation to enhance education with technology. Sure, whatever you say. ICT minister attends graduation ceremony of over 156,000 DEG trainees in 2025. What? Big number equals better. Look at these jack-offs. ICT minister, Microsoft MEA president discuss cooperation in AI capacity building. ICT minister, Yemeni counterpart discuss cooperation on digital transformation capacity building. Oh, look, there he is again. He's right there. No, yeah. See that guy? He's right there. Minister's corner, huh? Oh, hey, guy. What are you doing in the void? He looks just like Jared Fogle, doesn't he? What are you so smiley about? You're literally walking down a red carpet. Oh my god. Okay. Why does these buttons not work? Why do these buttons just not work? Why do they do nothing? Of course, first and previous doesn't do anything, but... even clicking on this doesn't work. That works. Nothing on your stupid site works. I mean, that's to be expected. You know? Not exactly... much intelligence going on here, right? But day 3 grand finale. You got the Billie Eilish fuck ass smile, but it's someone else other than that. And yum yum, delish, chicken. Helen Zepraz's. It's especially yum yum delicious because it's raw. Yeah, fucking raw. Yeah. Oh wow. Wow, yeah. Oh my god. This is such a freaky ass thing going on here. That's just it's just PNG, JPG. Wow. Oh, that's not even on the thing itself. Look at that. It's in front of it. Yeah. Which means this is too, and yeah. It's just images in front of the thing. And all that. Yeah. Oh, wow. Yeah. The waterfall model. What? How am I supposed to look at the waterfall model if you just... Like, you did that before, didn't you? Okay, and these are pins, right? There's pins in it. There's pins in the whiteboard. Tape. There are pins in the... Well, I mean, this isn't like whiteboard like this is, but... Even if I wanted to read about the waterfall model, guess what? I can't. Requirements, analysis, design, coding. What the fuck? I do nothing and it just does that. What the fuck? Why don't you want me seeing the waterfall thing? Why don't you want me seeing the waterfall model? Well, I don't think I even need to say how terrible design all of this is. How useless and worthless and incompetent and whatever, but... Okay. But hey, look, at least he's like staring right into your soul. Okay. Been reported in Xenopus human clawed frogs without necks and budding yeast. A recent report demonstrated the co-immunoprecipitation of 6 MCM proteins in Xenopus. Our results are consistent with the results from Xenopus and have clearly shown the physical association of NDA1, MCM2SP with all the other known MCM proteins in fission yeast. We're comparing frogs to yeast here. Yeah, that's what we've been doing this whole damn time. Or whatever. Something like that. The molecular weight of the complex prepared from... And, okay, it's consistent with it. The frog is consistent with yeast. That's wonderful. I'm so happy for you. What do you want me to... about anything. The molecular weight of the complex prepared from asynchronously growing cells is estimated by gel filtration to be 560 kDa. Okay, 560 kDa. We examined whether this value changes during the cell cycle. Data not shown. Total cell extracts were prepared from CDC10 mutant cells arrested in G1 or from cells arrested in S phase by hydroxyurea, and these extracts were loaded onto a gel filtration column. In either case, the elution position, right, the elution position of NDA1 and NDA4 changed little, if at all. Data not shown. This indicates that the molecular weight of the complex is the same, at least in the G1, S, and G2 phases. Data not shown. Note that asynchronously growing fission yeast cells are mostly in G2 phase. In Drosophila, MCM homologs have been shown to be fruit flies have been shown to be associated in a 600 kDa complex throughout the embryonic cell cycle. Similar observations have also been made in Xenopus. the purified MCM fraction contained nearly equivalent amounts of the 6 MCM proteins, figure 5, lane 8, which is the last one there, whatever. The molecular weight, I don't need to go back again. Okay, the molecular weights of the MCM proteins predicted from their amino acid sequences are in the 80 to 100 kDa range. It is worth noting, that NDA1 MCM2SP, MIS5 MCM6SP, and CDC21 MCM4SP show slower electrophoretic mobolites than their expected sizes. The purified fraction also contains 60 and 58 kDa proteins, P60 and P58, figure 5, lane 8, the same thing you're referring to again. We are not sure whether the first mention of we are not, we the people, we the Japanese, the dirty Japs, that should have been Hiroshima and Nagasaki, their grandparents. I'm not sure whether P60 and P58 are in fact associated with the MCM proteins, as the elution profile of these proteins from Superdex 200 might be slightly different from that of NDA1, NDA4, and the other MCM proteins, figure 4. The sum of the molecular weights of the MCMs and the P60-58 slightly exceeds the estimated molecular weight of the complex 560 kDa. We need to examine this issue further by other methods to detect molecular interaction. A determination of the amino acid sequences of P60, P58 will be informative. It has been noticed that the MCM protein complex dissociates at high ionic strength in other organisms. In the presence of 500 mm sodium chloride, the NDA1 and NDA4 proteins were dissociated and were... Wait, no, that's not... Is Na sodium? Fuck. I don't know why I'm just so doubtful. Okay, that's all. Yes. Yes, okay. I don't know. It could be other salts, right? Yeah. Wah. Okay, it is. Sodium chloride. It's sodium chloride. Sodium chloride. In the presence of blah, blah, 500 mm, whatever that's referring to, sodium chloride, the regular ass salt, the NDA1 and NDA4 proteins were dissociated and were eluted from a gel filtration column at 210 and 320 kDa respectively, data not shown. The extent of the sensitivity to high salt has found to be dependent on the type, salt on frog legs, they shake, dance a little, yeah, good stuff. Yeah, found to be independent on the type of anion. You do that with all kinds of things, they'll do that, but anyways, yeah. The type of anion, the MCM complex is more stable in buffers with acetate or glutamate than in those with chloride. Data not shown. See experimental procedures. The thing with that is too, it's like, you need a fucking working brain for this and that, but to be precise with things, to have precision behind things, to have purpose and meaning behind things more so than just it happening, it occurring. It can happen without that command or whatever, but it still has a command in some way. It's still what is commanding it? It's not something from even the cells, really. It's just that's what the machine was designed to do, was to not shake around a little, but the muscular stuff was designed for movement, so it moves. It does what it was meant to do. It does what it does. It's what it's for. It's for movement. So it moves if you agitate it a little. Anyways, the MCM complex is more stable if it's still relatively intact, which it is, then yeah. Complex is more stable in buffers with acetate, and then you only destroy whenever you go and cook it or cut it up sufficiently, but you really have to cut it up good and all that, but yeah, or just wait, obviously, right? Glutamate then in those with chloride, data not shown, see experimental procedures. It has been pointed out that acetate or glutamate could be closer to physiological conditions for intracellular proteins than chloride because the chloride concentration is low inside cells. Indeed, the replacement of potassium chloride by potassium glutamate enhances protein DNA interactions in vitro. Heard of that before. Vitriol. Vitriol is another word, but anyways. Just to make sure, Vitro is the largest glass producer in Mexico. Oh my god. Vitro fertilization. Yeah, meaning in glass. That's where I've heard it before. Yeah. Outside a living organism in a controlled environment. That's exactly what's going on here. That's all of what this is. So, yeah. But that's, well, of course, I mean, that's what it's referring, right? It's the different ways in which artificial insemination. But anyways, yeah, and that's just one of the things outside of the, whatever it is that they're referring to exactly with that. But, yeah. Outside of in your body. Yeah, it's inseminating the egg outside of, yeah. And then you stick it in afterwards or whatever, right? Et cetera, yeah. So that I can then from there and all that, you take the egg out, then you stick it into the egg, and then you stick the egg back in, the now fertilized outside of into the egg, and then it takes hold and everything from there, right? Like it was never even gone in the first place, like it's not a foreign body now or something, technically speaking, or whatever. I don't know. I mean, it's your damn egg. It's your stupid fucking egg, you ungrateful bitch. Okay. It is also worth pointing out that immunoaffinity chromatography was an essential step to purify the MCM complex to near homogeneity. Figure 5. For the high efficiency of immunoaffinity, see Zhao. Zhao. I will see Zhao. And Zheng. Zhao Zheng. 1992 and 1995. We estimate there are 3,000 NDA1 containing MCM protein complexes per cell, corresponding to one complex for every 4.7 kb kilobytes. No, I don't know. DNA, data not shown. I don't know what it is. This is approximately 10-fold less than the MCM2 protein in budding yeast, 1 MCM2 per 0.35. It is. Wow. Approximately. It's even... yeah, it's the other way around, right? Approximately almost, because I was thinking it looks like over if it's that, but it's almost if it's the other way around. And maybe closer to that in humans. 1 BM28 per 10 kb mention of BM28 mentioned, whatever that is. Yeah. The number of the MCM protein complex, it's just another one of these things, one of these same thing, MCM fucking right thing is another protein thing, right? But whatever. Still more abundant, but what exactly is it, right? What exactly is it? 5 to 10 fold. The number is still more abundant, 5 to 10 fold, than the number of replication origins estimated in fission yeast. It is also worth noting, however, that the portion of MCM proteins associated with NDA1 is not 100%. Under conditions where the NDA1 is quantitatively immunodepleted from the extracts, 30 to 40% of the other MCM proteins remain in the supernatants. Data not shown. While anti-NDA4 antibody beads completely deplete NDA4 from the extracts, only 70% of NDA1 is removed. It is therefore possible that several different types of complex exist which contain different sets and such or numbers of MCM proteins in vivo. Vivo! Vivo! Vivo, yeah. Our purified fraction may only consist of a subset of the endogenous MCM complexes. The association of 6 MCM proteins of 80 to 100 kDa molecular weight in a 560 kDa complex suggests that the proteins might form a heteromeric hexamer. One class of DNA helicases are known to be organized into hexameric rings, but we have so far not detected any DNA helicase activities associated with the purified fraction. The purified MCM complex has little affinity for DNA, data not shown. However, we may not be performing the assays under the optimum conditions for the proteins. Oh, you think? You might not be optimally doing anything, huh? You might be doing everything all backwards, huh? It is also possible that our purified complex is... I'm glad you admit it, though. It's also possible that our purified that's commendable. Purified complex is an inactive form since it has been prepared from asynchronously growing fission yeast cells that are mostly in G2 when the MCM proteins are supposed to be dissociated from chromatin and are inactive. It will also be interesting to examine the effects of protein modification, such as phosphorylation on the complex, and since in higher eukaryotes the MCM proteins dissociated from chromatin are highly phosphorylated. Another factor to be considered is the contribution of other initiation proteins. Several groups have recently shown the ORC and CDC 6 homologs are required for the loading of MCM proteins onto, separating in separate words, chromatin and xenopus. It will be important to study the interaction of the purified MCM protein complex with these initiation factors in vitro. Wonderful. The purified MCM protein complex has a globular shape, and its rotary shadowed images are often separated into several domains. Figure 6. Yep, we're referring back to that from before with the globular everything. Everything's so globular. Okay, this globular structure with several domains might be consistent with the oligomeric association of the MCM proteins that have similarities in their sizes and amino acid sequences. The boundary of the domains is often large and forms a cavity or a channel in the particle. Figure 6 DEF. I can recall that. The width and depth of the cavity at the center of the complex appears to vary between the particles observed. In addition, the numbers of the subdomains in different particles are not always the same. This heterogeneity in the structural details of the MCM particles might be induced during their preparation for electron microscopy, such as during the dehydration, absorption to a surface, or rotary shadowing. It would be worth investigating the use of other fixation/staining methods. It is also of interest to examine how the MCM particles interact with chromatin. Given the fact that the archaea possess putative homologs of MCM genes, the MCM protein should have a very fundamental role in the initiation of DNA replication. Bolt et al. 1996. We believe that a purification of the MCM protein complex will contribute significantly to an understanding of the molecular mechanism of the initiation of eukaryotic DNA replication. Lovely, wonderful. Yeah, okay, so it's from here. Experimental procedures, strains, and media. The construction of an S. Pombe strain, Ka25, that's you've been yapping about this whole damn time, huh? Reading about whatever it is, expressing NDA1 protein and tagged with a hemagglutinin epitope and 6 histidine residues has been described previously. Okay, so cells were grown at 33 degrees Celsius in yeast extract medium, 0.5% yeast extract, 3% glucose, supplemented with 75 milligrams each of adenine, I love adenine, and uracil per liter. DNA stuff, cloning and construction of an expression plasmids. A part of the MCM7 homolog in S. Pombe was cloned by PCR. Degenerated primers were designed according to the conserved regions of MCM7 proteins of S. cerevisiae, Dalton and white bread, and human. The nucleotide sequences of the primers are the following. Yada, 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 who gives a shit. Bases in parentheses represent mixtures. Bases, yeah. It's basic. It's not acidic. It's whatever. It's basic, yeah. Parentheses represent mixtures. Corresponding amino acid sequences are PEI, YG, and DEF, DKM, respectively. BAM-HI or ECO-RI sites were added at the not 5 foot, but 5 whatever, denoting this, symbol thing, ends to facilitate the cloning of the PCR products. S. Pombe genomic DNA was used as a template. The amplification conditions were 30 cycles at 94 degrees Celsius for 1 minute, 50 degrees Celsius for 2 minutes, and 72 degrees Celsius for 3 minutes. The amplified fragment of 0.33 kb was cloned between the BAM-HI and ECO-RI sites of P-BlueScript, Stratagene, and sequence. The sequence data of 0.33 kb fragment of S. Pombe MCM7+ are available from EMBL/GenBank DDJB under accession number AJ000065. A part of the S. Pombe MCM3 gene coxon was amplified by PCR and cloned as described above. The BAM-HI fragments encoding part of the S. Pombe MCM7+ or MCM3+ were cloned into the BAM-HI sites of the PGX vector to express GST. Well, vector, huh? Yeah. To express GST, glutathione, DNA stuff, fusion peptides in E. coli as previously described. Smith and Johnson, huh? Wow. something in parentheses mentioning something very recognizable, huh? Not just like some a GenBank too. That's familiar. Yeah, okay. Insoluble Smith and Johnson over everything. Big old conglomerate, huh? Anyways, a family company, they say. Yeah, but the whole family's dead and has been dead for quite some time, kind of. No, actually, maybe not. Maybe it's continued on by surviving, whatever the fuck. Family company, huh? Like, just that whole fucking thing at the end of each of those goddamn little ads, you know? A family company. So wholesome, huh? Right? Yeah. Insoluble fusion protein was isolated and gel purified. It's the whole shtick. It's the whole thing. Purified by diffusion as previously described. Watt et al. 1985. Adachi and Lamley 1992. Okay. Immunological procedures. Did I... Yeah, okay. Just making sure I got all that. Yeah, it's pretty basic stuff. Okay. Immunological procedures. The immunization and bleeding of rabbits. What? What? Why are we bleeding rabbits? Well, okay, with that wording, it's like, okay, maybe we're like taking samples of their blood or something, but the bleeding of rabbits, it just goes from whatever this and that to the bleeding of rabbits. What kind of demonic shit, huh? I'm into it. I like it a lot. Okay, the immunization and bleeding of rabbits was performed essentially according to the methods of Harlow and Lane 1988. 100 micrograms of gel purified GST fusion proteins were injected 3 times into each rabbit. The injections were performed with incomplete Froon's adjuvant containing 5 milligrams of poly A.poly U sigma, which means whatever you want it to mean, really, at any given point in time. Just they love to placeholder up the wazoo with everything. Everyone does, not just fucking nerdy individuals. As previously described, Hovhanessian et al., because meme shit too, right? Et al. 1988. For immunoprecipitation, the S-Pombe cell extracts were prepared in HNG buffer 10 herpes, sounds like herpes, hepes, salt and oxygen, hydrogen, or whatever, yeah. pH of 7.5, 50 mm sodium acetate. Well, I guess it's millimeters. Not millimeters. No, it can't be millimeters. I mean milliliters, right? But so it can't be. What the fuck is it then? I'm just getting things confused, but it's like, yeah, no, it can't be. What is it? Well, yeah, I mean, it's milli, but what is the large M? I don't know. What is... Shut up. Lowercase capital. I don't know. Like, you're just... Yeah. Science, whatever. When it comes to things that are case millimolar, millimoles per liter, there you go, a measure of concentration. Okay, fuck, with anything that's remotely based in lowercase capital shit, you just don't get a straightforward answer with anything. And I mean, I don't like it being that, like, fucking, I don't know, just regardless, it's just whatever. Millimole, yeah, or whatever, milligrams per, yeah, okay, in moles, and like I fucking any of that shit, I don't know, just, yeah, I've heard that plenty before, whatever. Okay, it's a measure of concentration, alright, per whatever, alright, okay, there you go, measure of concentration, this much per this much, alright, continuing on. Sodium acetate, 10% glycerol, plus 0.5, another measure of concentration, 0.1, I mean, millimoles, PMSF, phenyl, it's always phenyl methyl, whatever, phenyl methyl sulfonyl fluoride with glass beads. The extracts were clarified by spinning at 150,000 G for 1 hour. With the speed of light, like over 1% of the speed of light, like for an hour, like no, No, it couldn't be. I don't know. Just saying G, but whatever G is referring to, it can't be over 1% of the speed of light. Like, no, it couldn't be. It couldn't be. It couldn't be, no. Protein A Cephalose beads, Pharmacia, were loaded with the appropriate antibodies, usually 7 microliters of serum or 3 to 5 microliters of ascite fluid to 20 microliters bed volume. Washed and equilibrated. Let me look at this really quick. Hey, Dora the Explorer. Yeah. Okay. Okay, anyways. Last beads, the extracts were clarified by spinning for an hour. Protein A Cephalose beads, Pharmacia, were loaded with the appropriate antibodies, usually 7 microliters of serum or 3 to 5, yeah, I just read this, microliters of ascite fluid to 20 microliters bed volume. Washed and equilibrated with HNG plus 0.1 millimoles PMSF and 0.1% nonadate. It's probably nonadate. P40. Extracts containing 4 milligrams of protein were loaded with 10 milliliters, or not milliliters, microliters, I guess, or whatever. Yeah, it's small stuff, very tiny things. Yeah, spin them really fast, really nice and good that way. Yeah, of antibody-loaded protein Cephalose and incubated on a rotating wheel for 90 minutes at 4 degrees Celsius. The beads were washed 5 times with HNG plus PMSF and nonadate. I don't know. It sounds like a Japanese fucking name. It does. I don't know. P40, like WD-40. And the bound proteins were eluted by 30 microliters of sample buffer for SDS gel Lemli 1970. We found that the MCM proteins did not and now we're on page 10 of 13, we're getting there, did not co-immunoprecipitate efficiently in the presence of the 150 millimoles salt, regular salt, that is used in a standard immunoprecipitation protocol. Data not shown, Harlow and Lane, 1988. Switching the salt from regular salt to sodium acetate, 50 to 150 millimoles, greatly improved the well, that makes sense. I mean, usually just going from the default to something other than the default will yield better results. Greatly improved or worse, sure, but, usually it's something more in that kind of direction of nicer, typically, I think, on average, something other than the default. Default is just default. Default is lame. Default is boring and lame. Greatly improved the co-immunoprecipitation efficiency of NDA1 and NDA4, although the presence of a higher concentration, 300 millimoles of sodium acetate, reduced the efficiency to 50% relative to that seen with 50 millimoles acetate. Data not shown. Don't put too much in there. Don't put too much. Okay. Good to know, I guess. The extent of sensitivity to, just right. The extent of, maybe that's worth looking into, right? What is the optimal amount or something? I don't know if this matters nearly enough, but, right? What is optimal? What is the optimal amount of sodium acetate to be using? The concentration, I mean, right? The extent of sensitivity to high salt concentration. I mean, we mentioned this before multiple times, right? High salt concentration, not good. Not too salty, yeah. Not too salty, yeah. A lot of things. A lot of, biological stuff is very sensitive to salt. Yeah. Yeah. That's a pretty common thing, right? Not surprising in the slightest. The recovery of the complex was nearly 100% in the presence of 500 millimoles sodium glutamate. The presence of glycerol 10% also improved the co-immunoprecipitation efficiency. We did not detect any significant effects due to the presence of Mg2+, or EDTA in the buffer. Data not shown. Western blotting with anti-sera against fission yeast MCM proteins was performed as described. Anti-CDC21 and anti-MIS5 antibodies were kindly provided by Dr. Kiersey, University of Oxford, and Dr. Masukata, Osaka University, respectively. Anti-rabbit. Kill all the rabbits. Fucking golly. I mean, we're bleeding them, right? We're bleeding the rabbits. Anti-rabbit. We are anti-rabbit. Anti-fucking-rabbit. Rabbits are basically like rodents, you know. Anti-rabbit. Kill all rabbits. Fucking okay. All right. Anti-rabbit, IG, G, whatever, lowercase g, capital G. Well, we know what IG is, but then there's another G on there. What's another G? We just add another G on there. Who gives a shit? Antibodies conjugated, you know, just get over it. Whatever. Conjugated with peroxidase. That sounds familiar. Well, perox, you know, or whatever it is, certainly. But no, idase. And it's probably just the combining of those two together. But those two together I probably haven't seen in particular before. It's just those, like, alone by themselves. You know, DACE, peroxy, and DACE alone by themselves are like, you know, along with other things, but like together like this, peroxidase, I probably haven't seen before, or at least not enough to like recognize it due to that. It's just the individual things separated. I mean, okay, I think that makes sense. Were used as secondary antibodies and were detected by the ECL Western blotting system. Amersham is a sham. The chemiluminescence signals were detected by X-ray film or by a molecular imager, GS250, Biorad, GS250, whatever. Purification of MCM protein complex. Purification was monitored and quantified with immunoblotting by anti-NDA1 and anti-NDA4 antibodies. Ka25 cells were grown to 4 times 10 to the power of 7 per milliliter in 60 liters of Ye media supplemented with adenine and uracil. Very, you know, these are DNA things. These are, you know, the little letters that you'll find or whatever. That's what these are. There's only, you know, there's 4 of them for whatever, DNA, RNA, blah, 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 and they share 2 of them. So, like, there's 6 in total, or like 6 main ones or something like that. But then you have all these other fucking letters that you're throwing in too. So what's up with that? I don't know. But yeah, I don't know, just whatever, along with the sugar phosphate backbone or whatever of the spiraling ladder, you know. I don't know what's up with all the other letters or whatever that you're throwing in, but yeah, it's just like A, G, T, and was it N or something like that, and a couple others or something like that, because DNA, RNA, different things. I don't know. Something like that. Harvested by continuous centrifugation hereafter. So I don't know what's going on with all of that. I still don't know. But anyways, yeah. I'd like to know. I'd like to know what's with all the other letters that you're throwing in all willy-nilly. Way back there at the top of one of those pages, which wherever was it. We'll go back to page 10. This. I don't know what the fuck's with all of this, with all these million fucking other letters. Because, I mean, unless it's not referring to DNA stuff, right? Unless this is not, but like what the fuck else? I don't know, just a sequencing of proteins. Okay, I guess it's just a protein thing. I don't know. I'm not familiar enough with the nitty-gritty of all that, so I guess it just is a protein thing, like it's not a DNA thing, but I don't know, fucking whatever. Okay. I see a bunch of letters like that, of course I'm just going to think DNA thing. I don't know, whatever. Who gives a shit? Whatever. It's a sequencing thing, whatever, same damn thing, just of a different thing of it or whatever. Okay. I don't know exactly. It doesn't matter enough to me. That's how it is. I don't know because it doesn't matter enough to me. Where were we? Down here, right? Yeah. The Ye media supplemented with adenine and uracil. The cells were harvested by continuous centrifugation. Kisho, type number 4A1H1. Hereafter, all operations were performed at 4 degrees Celsius unless otherwise indicated. Good shit. Consistency, eh? No, I don't know. The cells were resuspended in 500 milliliters of cold HNG buffer. 10 millimoles, hippies, yeah. Whoa, where? Shoot them. Dead. God. Sodium, oxygen, hydrogen. What is that called? I've seen that before, plenty of times before. I don't know what it's fucking called, but let's see what it is. Also, this is called something itself. You know, it's is it a hydroxyl group? I'm not certain of that, if that's what that's called. It is. I got it right. It's a hydroxyl group. I was right. I'm so cool. I mean, you mentioned that before and all the live long day, but like connecting the dots there, I don't know. It is a hydroxyl group. Yeah. I know a couple of those. Yeah. I'm not good with names. I'm not good with labels. I'm not good with all of that. Yeah. Oh. Okay. So that's why it's so familiar. Fuck, shut up. All right, wait, what? Yeah, no, right. It's... Yeah, no, anyways, okay, whatever. Yeah, it's lie. Okay. The lie lies to me. Yeah, I know what lie is. It's just, yeah, okay. I'm not good with sticking two things together. Yes, I got the two things apart, but sticking the two things together, okay? Shut up. Sodium acetate, 10% glycerol plus 0.1 millimoles, PMSF, phenyl, fluoride, whatever. Centrifuged at only 1,000 G for only 10 minutes. The cell pellets, 380 G in weight, were stored at negative 70 degrees Celsius. Cells were disrupted by a bead beater. A bead beater, huh? Speaking of beating, huh? Speaking of beating, you are about to get a beating. Can't wait, no. With a 350 milliliter chamber, 90 grams of not 90 grams, what? 90 grams of cells? I guess, okay. Wowee, the scale is much larger here, huh? Hence why we're spinning things a lot slower and for less time, or whatever. Yeah. Okay. All right. Sure. Hundreds of grams. Is that really necessary? Okay. We're resuspend. Okay. 90 grams of cells were resuspended in the cell pellet, yeah, stored at negative 70 degrees Celsius. That's cold. That's really cold. Resuspend. Okay, 90 grams of cells are resuspended in 140 milliliter HNG plus 1 millimole PMSF, and 175 milliliters of 0.5 millimeter glass beads were added. This mixture was processed in a single operation. Extract/glass bead mixtures were transferred to 50 milliliter conical tubes with a pinhole at their lower end. These tubes were put in larger 250 milliliter bottles. Extracts were recovered in the larger bottles after centrifugation at 200 G for 2 minutes. There's no period there, but there should be. Cell debris was removed by centrifugation at 8,000 G for 10 minutes. 150S extracts were prepared by further centrifugation at 150,000 G. Oh, once again, returning to 150,000 G. Nice. For 1 hour. Beckman type 45 Ti rotor type, whatever, T. 80% of the NDA1 and NDA4 proteins were recovered in the 150S extracts. The 150S extracts, 400 milliliter, 10 grams of protein. Yummy. Were frozen in liquid nitrogen and stored at negative 70 degrees Celsius until you... If I have to see one more fucking time, liquid mentioned in liquid nitrogen. Like, there are so much other cooler ones. Yeah, but this is cheap. Yeah. I mean, it's the most abundant thing in the atmosphere. Yeah, it's cheap. It's cheap. It's good because it's cheap and it's easy and it's this and it's that and reusable. No, not reusable. Well, yeah, it's everywhere. Okay. Liquid oxygen, say, or hydrogen or whatever. Oh, you can't do a hydrogen because obvious reasons, but like, say, oxygen. It gets way too cold for that, liquid nitrogen, it's like just right or whatever. I don't know. It's just like especially cheap, really, is what it is. It's just cheap. That's what it is. It's just cheaper. So use that for everything, yeah. And stored at negative 70 degrees Celsius until use. 200 milliliters, 5 grams protein. I mean, I can't I don't know. Fucking grams of protein, huh? All right. The 150S extracts were thawed and supplemented with. What the fuck is with people taking protein supplement? Like, you can't just eat meat, you know? Like, just. Why don't you just eat meat? What is wrong with just. I really don't. I don't know. Just if you hate consuming meat but you love consuming powder, I guess. I mean, if there's like other things in there or whatever, but when it's just or some shit, I mean, it's all gross as hell is what it is. It's hootenanny shit, but like, just protein, huh? Protein, yeah. You know, it's probably just, you know, you probably have much better luck with like, you know, the actual utilization of that. The like, I mean, just the raw powder or whatever the hell, the very artificially this and that powder, it's like, it's just, I don't know, yeah. I don't know, it's just to sell you something, I think, is really what it is, but I don't know. I'm getting sick of eating chicken and whatever. That makes sense, I guess, yeah. All that time that you're spending on chewing meat, you could be spending pumping iron, you know? Yeah. Exactly. Right. You know, so just that and, you know, nice bite out of, you could get your MREs, right? You could get your meals ready to eat, or you could get your, you know, you can get your, yeah, what is it? Yeah, no, well, that's out of the question because that takes time. That's valuable time away from what you could be doing there, right? What was it? Yeah, what was I gonna say? Damn it, I don't know what it was. Yeah, um, yeah, I don't know, just insanity. Yeah. Oh, who knows? What was it? What was I gonna say? I don't know. Well, I mean, yeah, obviously, you know, like you're just holding it with your hand or whatever. Just it could be on a fork or something. I don't know. Yeah, and you just take bites off of whatever. You got your meat in one hand and you got your weight in the other, you know, and you just kind of switch back and forth between the two or whatever. Just kind of rub all the meat juices, right, as you like to refer to it as. Fucking gross as hell to say it that way, but you know, all along the all over the weights and whatever, you know, get it all nice and greasy, you know, it'll help you right, and people slather honey and what's it called, I don't know, some friction powder or whatever all over there. Yeah, all right, one sec. What? Are you hungry? Sure. Is it done? You want avocado? Sure. Where's the ketchup? Right there, but well, we have this ketchup too. On that. Want some grilled onions? Want some grilled onions? What do you want? Okay. Yeah. Okay, I got it. I can't see anything, you know? That's really, yeah. Oh, damn. Well, okay. We really shouldn't be using this. I guess as long as you don't get sick. How's your immune system? Okay. Wow, 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 wow. Okay. Hi, I have returned. Where were we? Sorry. I'm so very sorry. Oh no. Oh no. Oh no. Oh no. I have no idea. Okay, well, we were here at least. Okay, we'll continue right from here. Frozen liquid nitrogen stored at, yeah, until use. Recover in larger bottles, centrifugation. Yeah, sure, okay. 200 milliliters, 5 grams of protein, whatever, of the 150S extracts were thawed and supplemented with imidazole to 5 millimoles from a 1 mole stock at 8 in HNG. 4 milliliters aliquots were mixed with 5 milliliters of NTA. Anime speak NTA agarose quagen equilibrated with HNG plus 0.1 millimoles PMSF. The mixtures in 5 milliliter tubes were put on a rotating wheel for 2 hours. The resin was separated from the solution by spinning at 1000 G for 3 minutes in a swing bucket rotor, washed twice with HNG plus 5 millimoles imidazole. Oh, damn it. 0.1% nonade. Or I say nonade, sure. P40 and 0.1 millimole PMSF. 35 milliliters of the resin was transferred to a 2.6 centimeter diameter column and washed with 3 column volumes of HNG plus 20 millimoles imidazole and 0.1 millimoles PMSF. Bound proteins were eluted with 3 column volumes of HNG plus 200 millimoles imidazole and 0.1 millimoles PMSF at a flow rate of 35 milliliters per hour, I suppose. The elution of NDA1 and NDA4 proteins was monitored by SDS gel electrophoresis and Western blotting. Most of the histidine tagged NDA1, around 90%, was eluted with 200 millimoles imidazole. The peak fractions, 8 milliliters per 72 milligrams protein, were loaded onto a biogel, a 1.5 meter, I guess, I don't know, biorad column, 2.5 times, the dimensions of the damn thing, 67 centimeters, equilibrated with HNG plus 0.02% non-a-day, P40. 1 millimole, 2 mercaptoethanol, and 0.1 millimole PMSF. Okay, the flow rate was at 28 milliliters per hour, and fractions of 4 milliliter were collected. The fractions 4 milliliter, 3.8 milligrams protein containing NDA1, NDA4 proteins were combined in a 50 milliliter tube. 0.3 milliliters of protein, agarose, Gibco, whatever, which had been loaded and cross-linked with the monoclonal antibody 12CA5, 10 milligrams, milligreeters, huh? 10 milligrams IgG per ml bed, anti-HA beads, Harrow and Lane, 1988, was equilibrated in H&G plus 0.1% non-a-day P40 and 0.1 millimoles PMSF and added. The tube was placed on a rotating wheel for 15 hours. The anti-HA beads were recovered by centrifugation at 1000 G for 3 minutes. That's not much at all. Washed 5 times with 40 milliliters of HNG plus 0.1% non-a-day P40, and then washed twice with 40 milliliters of H-Glug. I don't know, H-Glug-G? H-Glug. Alright. Sure, why not? 10 milliliters, or millimoles, hippies, lye, pH 7.5, 500 millimoles sodium glutamate. Damn. Wait, lye can be at a pH of 7. Like, that's weird as hell, you know? Like, what the hell? Yeah. What is that? That's nothing, I guess, yeah. Okay. Whoa. Okay, glutamate, 10% glycerol, plus 0.02% chaps. Chaps. Well, old dear old chap. Eh? Chaps. Chapstick. Yeah. And then it's used as a generic term, just like Band-Aid. Right? Good stuff, yeah. That's how you know you really made it, when your goddamn brand name is used for every other fucking thing that's the same damn thing. Wow. The resin then was transferred into an Eppendorf tube, and 300 microliters of 1 milligram per milliliter of the 12CA5 peptide and H-Glug plus 0.02% chaps was added. The tube was incubated at room temperature on a rotating wheel for 30 minutes. The supernatant of the resin was recovered. The incubation was repeated with a fresh peptide solution, and the supernatants were combined. 45 micrograms of the MCM protein complex was recovered in the 600 microliter of the peptide eluate. Yeah, just like those things. Eluate. The efficiency of the elution from the affinity column was estimated to be 20% under these conditions. Lovely. ATPase, and DNA helicase assays. Reaction mixtures 20 microliters of the ATPase assay contained 10 micromoles heat page lye, pH 7.5. 10 micromoles magnesium acetate, 10 glycerol, 10 micrograms per milliliter each of lupeptin. Yeah. Lupeptin, yeah. Oh no. My water cup. Fuck. We are fucked. Okay. Where were we? I don't even know. Oh no. Lupeptin, right. Lupeptin, right? There, yeah. Okay. Chemostatin, pepstatin A, and antipain. Okay. Antipain. 1 times protease inhibitor cocktail. 0.2 micromoles ATP, 3 micro CI, whatever that is, gamma dash 32 P ATP, fucking greater than 5,000 CI per mmol. Amersham 500. Oh my god. NG. What even is the N? No, what is the N? No, I'm not gonna just ask him then. What is NG? Velar nasal consonant nanogram. Right. Nano, of course. Well, I mean, that's what it means with fucking, yeah, it's nanometer, so nanogram. Well, of course, it's just... I'm tired now, okay? And I'm focused on food stuffs too. Okay. Okay. 500 nanograms, yes, of course. Step lower than micrograms, of course. Yeah, 1,000th or whatever. So it's half of one microgram. 500 nanograms is the same thing as 0.5 microgram. Yeah. Anyways, 500 nanograms of purified MCM complex, 2.5 micrograms of DNA was added to some samples. Good stuff, yeah. The mixtures were incubated at 30 degrees Celsius for 1 hour. Reactions were stopped by adding EDTA to a final concentration of 25 micromoles and cold ATP to 5 micromoles. Millimoles, sorry. Millimoles, and then this is, right, again, 1 microliter. Well, it's because they both start with M, okay? And yeah, I'm saying them back to back, and it's like, it doesn't matter. It does matter, but it matters in the context of you performing all this stuff, but it doesn't really matter all that much outside of it. It's just whatever. Specificity, exactness, oh my god. And reciting things, it's right there, okay, whatever. 1 microliter of the samples were spotted on a polyethyl aniamine plate, dried and chromatographed using 1 mole formic acid, 0.5 mole lithium chloride. Tell me about lithium chloride. Has a role as a geroprotector. Oh, sure. This is, oh, wow. Well, I mean, this is cooler than Wikipedia. That's for sure, right? Or is that? Has a role as a geroprotector, MNR, chemical shift reference compound, and an anti-manic drug. Yeah, they do use lithium stuff for that. You know, take this lithium thing, yeah. Whatever. It's a lithium salt and inorganic. No, actually, I have heard of lithium chloride specifically for that. Yeah, yeah, anti-manic, yeah, that thing, yeah. Yeah, that thing, yeah, right. So we're using that for this, you know, the fucking mental med thing. I have heard of, yeah, that. Lithium, yeah, thing. Which, I mean, well, either of them by itself, you know, but together they're pretty stable. Ah, fuck. Okay. The distribution of the radioactivity was examined by a molecular imager. GS 250 Biorad. DNA helicase assay was performed using substrates of a forked structure with 14 nucleotide non-complementary tail at the 5 whatever the fuck end, or at the 3 whatever the fuck end. Budd and Campbell, 1995. 10 pmol of the oligonucleotides were labeled with T4 polynucleotide kinase. And is that gamma? Is that the gamma symbol? Well, it's something like that. Is that gamma? How the fuck do you get G from that? What? Okay, just show me gamma. Yeah, it's gamma. Cool. Gamma 32P, ATP purified by ethanol precipitation and annealed with M13. We're on page 11. Don't worry, we're almost there. I don't know why the fuck would anyone sit through all this, huh? Boring as hell. Bovine serum albumin. I don't know. M13, MP18, single-stranded DNA, and 20 millimoles tris hydro... whatever the fuck... hydrochloride or something. It's called... I don't know. Fuck. It's just... Okay. pH 7.5, 10 millimoles... fucking... Is that mercury? Is that what Mg is? Or is it like fucking potassium or some shit? I don't even fucking remember. Shut the fuck up. Magnesium? Yeah, that makes sense. Whatever, it's something. Oh god. God damn it. Okay, magnesium. Chloride 2. Fucking, they got multiple of the same thing. Yeah. God. 100 millimole fucking regular ass salt and 1 millimole DTT. DTT? What's DTT? Fucking. It's just some random thing. Potent reducing agent. Okay, sure. Yeah, it's an organosulfur compound with the formula blah blah blah. Colorist compound is classified as a dithiol and a diol. Ah, dithiol and a diol, of course. A type of organosulfur compound with two thiol SH functional groups. Their properties are generally similar to those of monothiols in terms of solubility, odor, and volatility. May class. Sure, yeah. Glycol. An aliphatic diol may also be called a glycol. Saturated. Can be saturated, in which all the carbon-carbon bonds are single, requiring the structure to be completed or saturated by hydrogen, like hexane, or unsaturated, like hexene and hexine. Open chain compounds, whether straight or branched, and which contain no rings of any type, are always aliphatic. Cyclic compounds can be aliphatic if they are not aromatic. Ha 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 ha! Aromatic! Oh my god! What? Aroma is breathing it in. What is this? What is this bullshit? Don't you fucking turn aroma into meaning something else, you stupid mother... The first known use of the word aromatic as a chemical term. Like, it's like... I don't know, that it... what's the word for that? Yeah, volatile. I don't know. It lets off fumes. You know, which, I mean, I hate the whole multiple meanings with the same fucking word. Shut up. God damn it. God damn it, yeah. Ah, oh, damn it. Yeah. I got a sandwich right here, okay? You know what's so much fucking cooler than aromaticity? Sandwich, yeah. Holy hell. Holy hell, yeah. Okay. Whatever. Sure, that thing. DTT by heating at 98 degrees Celsius for 1 minute. 65 degrees Celsius 1 hour and cooling slowly at room temperature several hours. The oligonucleotide M13 hybrid was separated from non-hybridized oligonucleotides by gel filtration through a 1.5 milliliter Cephalose 4B column, equilibrated with 10 millimoles tris hydro. Is that hydrochloric acid or something? Is that what, yeah, HCl, right? Yeah. Yeah. Hydrochloric acid, right? It's that. I just fuck. I don't know. I haven't been looking. I mean, really, yeah. I mean, I haven't been looking at this kind of stuff for a while now. Okay, just in general, and I don't know. And I'm tired, and I'm this, and I'm that, and I'm blah 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 now. But I just reasons, not excuses or whatever. Disappointing, kind of thing with myself, I guess. I don't know. The reaction mixture, 20 microliters, contain 10 millimoles HPs, lye, pH 7.5, 10 millimoles magnesium acetate, 10% glycerol, 1 times protease inhibitor cocktail, 100 micrograms milliliters, BSA, bovine serum, albumin, sigma fraction V, 4 millimoles ATP to times 10 to the power 4 CPM, around 15 F moles, fucking substrate DNA, and a lot of this stuff is the kind of stuff that you just kind of skim over in your head, you know, like you don't fucking read this shit out loud either, you know what I mean? It's just a bunch of bleh. But I'll read it out just for you or something, I don't know. 500 nanograms of purified MCM complex, 400 nanograms of bacterial single-stranded DNA protein was added to some of the samples. The reaction mixtures were incubated for 20 minutes at 33 degrees Celsius. The reaction was terminated with 0.3% STS, 20 millimoles EDTA, 0.04% bromophenol blue. That's what we had way before. Mention of bromophenol blue. Yippee, the different kind of blue. That's not the same thing. Remember, that's not the same thing. Brilliant blue, whatever, is not the same as bromophenol blue. The samples were separated by electrophoresis and a 12% non-denaturing polyacrylamide gel. The gel was fixed in 10% methanol, 10% acetic acid for 30 minutes, dried under vacuum and autoradiographed. Low-angle rotary shadowing electron microscopy. We're nearing the end, you know. The purified MCM protein complex was dialysed against 10 millimoles HPs. What is this, anyways? What is this herpes shit? Anyways, lye again. What is this herpes? What are you, herpes? Sulfonic acid buffering agent. Yeah, one of the 20 goods buffers. Cool. All right. Compared to bicarbonate buffers commonly used in cell culture, record an unwanted photochemical process wherein it catalyzes a reaction with riboflavin when exposed to ambient light to produce hydrogen peroxide. This is not a problem in bicarbonate-based cell culture buffers. It is therefore strongly advised to keep solutions containing both HPs and riboflavin in darkness as much as possible to prevent oxidation. Oh yeah, things like oxidation. Wow, I mean, you know, that kind of stuff sucks, huh? How do I deal with that? It wants so badly to be oxidated, and then, well, at least then it's a protective layer until you want to cut it off next, right? That's not always the case with everything. It is a lot of the time, most of the time, probably, right? At least with things like metals and whatever, right? Protective outer coating can't penetrate further, but yeah, I'd imagine with like lighter things, right? Where it can seep through. Like, yeah, with metal, but metal is metal, you know? Not all too much is going to get through metal, typically. But, if it's undamaged and all that, but in the case of, what have you, lighter stuff or whatever, less dense stuff, you know, with less weaker bonds that don't, you know, outgas as easily or outgas more easily or something like that, right? You know, vaporize and all that, and you can smell it and whatever, all that, because the whole thing with metal is, you know, you only smell it off your hands because of the way it you know, your skin oils and whatever, gross as hell, but you know, interacting with it, that's what you're smelling, the interaction with that and all that, and that's yeah, okay, that's cool, I guess. Anyways, but yeah, I'm sure, you know, that's not the case with everything, right? It's just most stuff, at least, you know, pretty much most stuff, if not all of the metals, not all of them, right? There's weak as hell metals too, so, you know, let's not, you know, whatever, or soft at least, or whatever, right? Or highly reactive or not, or, you know, yeah, sure, gold, but not really. Yeah, I mean, yeah, well, lithium, sodium, whatever, those are technically metals. But yeah, you are consuming metal and rocks all the damn time. You're made out of metal and rocks too. Wow, amazing. And dialyzed glycerol, whatever. The sample droplets were placed on mica and dried dried at room temperature in a vacuum at 10 -8 millimeters. This is mercury, right? Fuck you, oh my god. Yeah. In freeze etch equipment, Hitachi HR7000 for 10 minutes. Dried specimens were rotary shadowed with platinum using an electron gun positioned. Oh my, it seems a little overkill. No, it's nothing really. It's just calling it that because it sounds cool and whatever, and that's what it is, you know, like a gun, yeah. Pew pew. You know, it's just like a laser on it, basically, but it's electrons and a laser, like, it's nothing special to it. It's nothing. It's just higher powered flashlight, more focused, right? Is there anything about a fucking flashlight? No. Well, it's just a higher powered, more focused flashlight. In the case of electron gun, you know, same thing, just with electrons, right? Yippee instead. Positioned at 2.5 degrees to the mica surface and then coated with a film of carbon generated with Formvar film. The specimens were observed with a GEM 100CX electron microscope. Yeah. Well, let's take a look at an electron gun. Why don't we? What does it look like, eh? Electron gun inside a TV. Oh, sure. Sure, yeah. How does it work? Less so how does it work, what does it do? What's its purpose, right? How it works, yeah. Star Wars, blah, 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 yeah. CRTs, right. This is why, yeah. Why do you know nothing about it? Because it's in CRTs and I don't give a shit about CRTs. Oh, okay. Big glass tube is used in most television and computer monitors. Electrons get aimed at the screen where they light up the phosphor in the screen to create the image. Is it black and white TV has a mention of black and white TVs? God, black and white TV has a single electron gun, while a color TV needs 3 guns because each pixel on the screen is made of a red, a green, and a blue dot. You cannot be like... Do modern monitors use electron guns? Like, they have to be very small, huh? Good bit smaller than that. It's like, yeah, I mean, it's like, yeah, I mean, you're saying CRTs, but it's like, then you're like saying like all that. What the fuck? When is this from? Like, I need a date on this. I don't know. Just used in. Oh, cathode ray tube, right. See, yeah, that. No. Yeah, that's what CRT means. I'm tired. Fuck, what the fuck are you even... How old is this? Whoa. What if I shot my TV... Nothing much would happen, really. Mention of the electron gun. So this is what TVs still look like in your mind, huh? I mean, hey, when I was a child, you know. Yeah, but... even still then and all that, but like... And, you know, you'll find it in the fucking mental hospital, all that crap, but like... and just at school and whatever too, but anyways, every now and again, and you know, stick the VHS tape in and whatever, yeah, all that good stuff, you know, well after the fact, but still, I don't know. Anyways, every now and again, as you're driving down the freeway, you'll see that famous bumper sticker that says "Shoot your television." Many times as you're watching television, the thought of a well-placed shot may cross your mind. I mean, if you want to, go right ahead, I guess, you know. But, like, okay. Whether it's a bad game for the home team, a bad sitcom, or a bad commentator spouting off about something. Bad. Shoot. That's how it works, yeah. There are lots of reasons to kill the tube. The tube, huh? What if you did actually take it out back and shoot it? We are, of course, talking about a standard television with a huge glass picture tube. Standard television, huh? We are living in the past, huh? Alright. Standard television with a huge glass picture tube. Standard television, yeah. So there's something big and meaty to aim at. TVs in the 25-inch and greater range have a massive piece of glass that weighs between 50 and 100 pounds. Yeah, sure. Here at How Stuff Works headquarters, we actually tried this experiment. Since it is a vacuum tube, there's been a lot of discussion in the urban legend community about a massive implosion that would occur when the bullet first cracked the glass. What are you talking about? The idea is that the vacuum would suck the glass fragments in, and then they would rebound at shrapnel speed. Sounds exciting, but no. At least when we try to hear nothing like that happen. No, of course not. Why would it? How fucking powerful do you think this goddamn vacuum is? You know what I mean? It's like... It's not a bomb waiting to go off or anything like that. It's just... My God. At least when we try to hear nothing like that happen, obviously nothing like that is going to happen. It's like the most that you're going to get is like smoke or whatever, you know? From it or something like that. But anyways, the bullet, you know, what you'll get from the regular ass electronics too, it's like the bullet went in very cleanly, punched a very neat hole through the glass, and air quickly filled the tube through that hole. There certainly was not an implosion. Will TV ever be the same? After you've taken the shot, there's no good reason not to take the whole thing apart and explore the interior. So that's what we did. A hammer cracked away the rest of the glass, and here's what was inside. TV components. Who knew? The front glass. Front glass is an extremely thick, sturdy piece of work. It's really high quality. It's actually leaded crystal, like optical glass, to give it great clarity and consistency. Suddenly, this turned into an advertisement for nothing in particular, just TVs in general, I guess. What the fuck? The front piece contains between 1 and 2. Who are you working for? You know, just TV industry in general, huh? Contains between 1 and 2% lead. The phosphor on the back of the glass, which is fucking nothing at all, but you know, leaded. It's like, well, I mean, it's good to mention it. It's just like, I don't know, whatever. The phosphor on the back of the, you know, and that whole thing when it comes to like radioactive stuff. I mean, it's not like it being see-through at all is kind of a thing, but like, yeah, you know, and whatever. It's just, you know, blocking out the higher frequency stuff, not necessarily the visible and weaker stuff, right? It's just, oh yeah, radiation protection stuff, but more so than leaded, you know, heavier elements than that. And the whole thing with lead usage to block radiation is just, well, it's only good for some, you know what I mean? You want, right, like water, right? You want, when it comes to, you know, when it comes to fusion stuff, you want lighter stuff. When it comes to fission, which is pretty much everything ever, then you want heavier stuff. Yeah, and you want heavier stuff even than lead. It's just lead is cheap, so lead gets used for everything, and then it becomes the whole thingy thing, standard or whatever for anything. And even then, apparently, like, oh, because of, like, say in a dentist office, because of all sorts of whatever, this and that, apparently, so they say, whatever apron shit is, it's like barely anything above background, you know, it's like eating a banana or whatever the fuck nowadays or something, a good bit of the time, I guess, supposedly, or whatever, but, you know, accidents happen and all that, and, well, I don't know, I mean, it's just like over your chest, haha, it's just localizing it, it's like, oh yes, well, you know, to the teeth, to the jaw, got a couple of, I think, cracks in my teeth, by the way. I don't know, my two front teeth are fucking, you know, two front teeth get a lot of usage, and they got, yeah, chips and whatever. It's good stuff, right? Yeah. I hate being alive. Front piece contains between 1 and 2% lead. The phosphor on the back of the glass is a phosphor coating. It's a white powder that flakes off. The shadow mask right behind the screen is the shadow mask. You don't need the mask in a black and white TV, but in a color. How fucking old? God. In the color TV, you need it because there are 3 electron guns and 3 different colors of phosphor on the screen. At each pixel on the screen. Like, is this from the 70s or something, all of this shit? Like, what the fuck? No, TVs looked more old-timey than this in the 70s, so it couldn't be in the 70s, but like the 90s or something. Even in the 90s, it's like you have this, but at the same time you have mentions of fucking black and white TV. It's like, okay. Hey, black and white TVs existed at one point, like, and things associated with it. It's just, okay, I don't know. You know, the mention of it, it's like cool to make the mention of it, the distinction in this and that, but like, you're saying it like it's like still in very much so in high usage, you know? Like everyone ever is like, half of everyone has a color TV and the other half still has black and white TVs, you know? Like you're saying it like that, you know? Like it's 50-50. I don't know. Everyone who has a TV, you know, half is black and white still or whatever, but it's like, I don't know. Yeah. 3 electron guns and 3 different colors of phosphor on the screen. At each pixel on the screen, there are tiny dots. Yep, red, green, and blue phosphor, and the mask makes sure that the right electron gun aligns with the right dot. Most common way to make the shadow mask is to take a thin piece of metal and punch hundreds of thousands of incredibly tiny holes in it. Yeah. The electron gun. At the back of the tube is the electron gun. Once you chip it out, it's a very elegant looking piece of metal and ceramic. Okay. 3 things happen in the gun. Filaments in the back of the gun heat up to produce the electrons, and the electrons get accelerated, and then they focus into a type beam. Where have I heard that before? When the electron beams, 3 of them in a color TV leave the electron gun. The electrons are moving at about 1/3 of the speed of light. That's slow as hell, really. Case of electrons, yeah. That gives them enough energy to light up the phosphor when they hit it. Sure. So that's what would happen if you actually shot your television. It's probably not an experiment that you need to repeat, because what you end up with is 50 pounds of leaded glass fragments all over the yard, and it makes a mess. A big mess. And it makes a big mess, yeah. Causes the faint horizontal lines on my monitor. Most likely you have purchased a cathode ray tube monitor based on Sony's trinitron technology. These monitors use an aperture grill. Having one significant problem. This is what causes the horizontal lines you see. Or the 80s or something, like Sony's trinitron technology. Okay, well, we got a mention of something in particular, right? Yeah, I mean, yeah, no shit, past tense. Fucking. This really is an old ass article, yeah. I don't know, yeah. Ended production in 2008. Well, it's at least prior to 2008 or whatever. At least, you know. Britney Spears voluntarily checks into rehab facility. Okay. All right. Sure. Yeah. Dot pitch. Oh yeah, tell me about dot pitch. What was that with how television works with the fucking dog and everything? Yeah, just like that, but with. Oh my god. Okay. Well, anyways, enough of all. Oh, that says a date on it. You know, these other things don't say a date on it, but this says a date on it. Updated. It doesn't say date. It says updated. March 5th, 2024. God, March 2024, huh? Oh, fuck. Anyways, television is certainly one of the most influential forces of our time. Wow. Really great waste of time, huh? Through the device called a television set or TV, you are able to receive news, sports, entertainment, information, and commercials. The average American spends between 2 and 5 hours a day glued to the tube. Nobody calls it the tube. Nobody calls it the tube. What the fuck? You ever wondered about the technology that makes television possible? Me. Pixelated image of a little boy. Is that what that is? Yeah. Angular gyrus. Yeah, good stuff. How telescopes work? Well, they got all these glass things and lenses, and yeah. Telescope, yeah. Telescope, yeah. How fabric displays work. How is the LCD in a laptop computer so bright? Yeah, how black lights work. No, don't use the black light. Don't turn on the black light. No, please, not the black light. No. An electron gun and this and that, and it looks kind of like a minigun. Wow. But it's just a tube thing. It's just a metal tube. It's just a boring and lame. Anyways. Let's get through all this. This is becoming such a chore. Okay, the sample droplets are placed on mica and dried at room temperature in a vacuum at -8 millimeters mercury in freeze etch equipment. Hitachi HR 7000 for 10 minutes. Dried specimens were rotary shadowed with platinum, used an electron gun position at 2.5 degrees to the mica surface, and then coated with a film of carbon generated with FormVar film. Yeah, the specimens were observed with an electron microscope. I think I just read all that, yeah. Anyways, other methods. The DNA manipulation was performed by standard procedures. SDS gel electrophoresis was performed according to Lemley. Protein concentration was estimated by the method of Bradford or by scanning the SDS polyacrylamide gel stained with Kumasi blue R250. BSA was used as a standard. Acknowledgements. We thank Dr. Domenico. Oh my god, credits, roll credits. That was it. That's the end of it. I didn't expect it to just end like that. Okay. We thank Dr. Domenico Majorano and Dr. Stephen Kiersey, University of Oxford, for the anti-CDC21 antibodies. Dr. Hiseo Masukata for the anti-MIS5 antibodies. Dr. Keiko Udaka is acknowledged for her generous experimental instruction in immunological procedures. We thank Dr. Stuart McNeil, Dr. Kevin Hardwick, and Dr. Jeremy Brown for comments on the manuscript. This work was supported by the Ministry of Education, Science, and Culture of Japan. Wonderful. References, a bunch of identification of nuclear fucking... a bunch of references. Yeah. Yeah. Cool. Very, very cool. Very, very cool. All right. Yeah. And then it continues on and on and on and on and on and on and on for the rest of all of it. And yeah. So that was something, right? Did you learn something here today? I don't know. 420. Okay, we gotta end it at 420. We can't go into 421. 420, yeah. Okay. Love you. Bye bye.